and poster presentation is going on, as well as the offline poster and oral presentations are going on. The keynote speaker for this session is Professor Dr. Mukesh Jain, JNU, Delhi, and the session chair is Dr. Gyan Prakash, Head, Seed Science and Technology, IARI, New Delhi. To the dais, I welcome Dr. Mukesh Jain, sir, Professor and Dean, Life Sciences, Jawaharlal Nehru University. He has worked extensively in the areas of plant genomics, biotechnology, and computational biology. He is one of the pioneers of next generation genomics research in plant sciences in India. For his outstanding scientific contribution, Professor Jain has been honored with several awards and fellowship from the national academies. Dr. Jain's current research interests include understanding epigenetic and chromatin level regulation of abiotic stress responses and seed development in crop plant using integrated multinomics, single cell genomics, and systems biology approach. He has published publication in national and international journals. He is fellow of National Academy of Science, Prayagraj, and also a fellow of National Academy of Agricultural Science, New Delhi. He is also an associate editor in various national and international journals of repute. I welcome you, sir, to the 8th AMIFOST 2025. I request our dignitaries to join us on the stage. I request Mukesh Jain, sir, Gyan Prakash, sir, to please join us on stage. I request Mukesh sir to kindly join us on stage. Sir. Sir, we have your PPTs. Okay. I think we don't have the moving mic which you requested for. Yeah. Thank you, sir.
That will be better, I think. I don't know why. That will be better.
लाइट चेंज चाहिए ना आपको?
नहीं, पॉइंटर चाहिए.
पॉइंटर हाँ वो भी
स्लाइड तो यहाँ से चेंज हो जाएगा.
मैं वो भी अभी लिखा था. थोड़ा भी वॉल्यूम थोड़ा देखना पड़ेगा वो बढ़ा दो.
Very good afternoon to all of you. Am I audible? Back. Is it clear? Good. So already I have been introduced. Thank you very much to the organizers for giving me the opportunity to be here talking about some of my work. So I come from School of Computational and Integrative Sciences. The name of the department looks like I do only computational work, but my background is molecular biology and biotechnology. So initially, I will be giving you an overview or introduction that why genomics approaches are important for the food security. I will try to convince you, and then I will give some idea about the kind of work we are doing that is supposed to basically enhance the crop improvement, and we have some examples also which can ensure the food security. So we all know that today's scenario we have a lot of challenges, specifically for our agriculture or ensuring the food security. the most important challenge is the climate change. We all know that global warming is happening, temperature is increasing day by day. Every year is being coming as the warmest year for last 10 years or so, except maybe 1 or 2 years in between. Then overpopulation, we are going to be 10 billion by 2050. And you know the India tops among all the countries. Then another challenge is the malnutrition. So even if we have food, how good is the food? So malnutrition can be overnutrition or undernutrition also. Then another challenge is because there is a huge disruption in the food supply chain. So all these together actually are threatening our food security. Now, again, I have put this question. So how we are going to feed 10 billion by 2050? And this challenge is even more important for India because we know obviously we are the most populated country and our bowl seems to be empty. So we have less resources. Maybe there is a minor increase in production, but at the same time, we are swelling in our count in terms of population. So, of course, we need to take some urgent steps so that we can ensure food security for everyone. So now here is the cartoon. So there is a huge crop diversity. I am sure just for the students' sake, what does the crop diversity mean? So you go to the market, you see there are different types of, in any Kirana shop, you will see different types of rice. Maybe 10, 20 types of rice you see there. You go to the any other kind of dal or anything you buy. So you will see different types of that, small seed, large seed, longer one, smaller one. So similarly for rice, we even within India, we have more than 1 lakh varieties available in our Gen Bank, which is there in NBPGR. Same thing is for other crops also. So every variety, every such cultivar have a particular character. Like just I mentioned, larger seed size, smaller seed size. Some may be good in nutrition, other may not be good in that much nutrition, but they are high yield. Or there may be something like they have some specific iron content or those kind of things. Some of the varieties can grow with less water, some varieties need more water. So we have such a huge diversity available. So if we can employ certain approaches, that diversity, that good characters of different varieties can be harnessed so that we can have sufficient food. We have improved crops, which we are growing regularly. But out of those 1 lakh, say I mentioned for rice, there are only maybe 1,001 which are being grown. Right? So it means that we are not harnessing fully whatever germplasm or where diversity is available with us. So here in the center, I have placed the genomics. So there are approaches available that we can imply on those crop diversity which is available, and we can select the good characters. When I say genomics, so good characters in the form of the DNA sequence which is corresponding or which is responsible for that, those good characters, if we can bring those into the varieties which we are growing heavily in different areas of the country, so we can have a crop improvement, and that can ensure the our food security. So there are different approaches, starting from the cross breeding, which I think we are doing for last several 100 years. So there are two parents and they are being crossed, and then there is next generation, then next generation, and then maybe you can get some good characters in the following generation. Then mutation breeding, so it's a kind of like you create certain mutations, you expose your plants with certain gamma rays or some other EMS mutagenization, certain chemicals, and then you get mutations, and then you do the screening. And you may get certain seeds which have the desired characters. So both of these technologies, they take a very long time in development of a new variety which has the desired characters. It can be up to 10 years, 15 years. Then transgenic breeding. I am sure transgenic word most of you might have heard. So when we bring some foreign DNA inside existing individual, so that becomes the transgenic. So, but it is not acceptable because of several ethical reasons. Although we have been doing this transgenic work for past, I think, now 3-4 decades, but still no product has come out, at least in India, based on transgenics because of the regulation. Now, recently, we have moved to the genome editing. Again, I'm sure many of you might have heard about the CRISPR-Cas technology. So that comes under the genome editing. So that gives a flavor that you can edit the gene inside the DNA of an organism without incorporating any foreign DNA. Recently, Government of India has given a clean chit in terms of the ethical regulations. Of course, not all types of genome editing, so SGN1 and SGN2. Maybe if you want to read, you can read more about that. So, the time taken by the genome editing technology, it has reduced to up to 3 to 5 years for development of any new variety which has the desired character. But what you need for any of these, especially the transgenic approach or genome editing, you need to know which region of the DNA you have to target. So how you identify that particular region? So it might be a gene or it might be any other sequence within the DNA, because there are so many regulatory elements. So how you can identify? So that is the challenge. So of course, at the center of all these technologies are the genomic resources. You should know what is the sequence of the DNA, what they are transcribing in the form of the genes, what are the genetic maps. So if there is association between a gene or a trait or even an allele or a trait. Now, how to select the target genes? So that's what I have already pointed out this question. Now I have mentioned here signature rather than a gene, because so far we have been focusing on the genes only, the protein coding genes. But it is not only the genes, these are several other DNA sequences, especially the regulatory sequences, that also play a major role in determining any phenotypic character. So even those can be targeted. So here I have put a kind of diagrams at the how the DNA is packaged. So it can be higher order, starting from the chromosome till it is being transcribed, that I have shown. Even after transcription, there is a translation and then metabolite production. So actually, any of the signature starting from the chromosome till it is being transcribed, so that can have a role in determination of any kind of phenotype. Even a single nucleotide change, even if the sequence is not changing, some epigenetic modifications, that can also help. So even how they are bound around the nucleosome, I am sure many of those who are from the biology background, they understand this. So how the chromatin is packaged, so that also determines many of those phenotypic characters. So what I want to say is, whatever is the phenotype, whatever you see, it's all in the DNA. You know, nowadays, especially after the COVID, now everybody knows about the DNA. So everything what we see, it is somewhere, something is coming out of the DNA of an individual. But most of the traits we are talking, like a plant growing on with less water, means under drought, or a plant growing under the salinity, or a plant having a larger seed size, these are all very complex traits. It's not very straightforward that just a single sequence or a few nucleotides, they themselves or they alone are controlling those. These are very complex traits, and it's a big challenge to identify those sequences or those particular signatures. It's something like that you are diving into an ocean. So this is a figure I have taken from a recent review in Trends in Plant Science, which have summarized that how the genomics approaches can be used. So on the left hand side, you see there is it is representative of all kind of diversity, which I just mentioned, which is available. Then you can employ different kinds of omics approaches, DNA sequencing, multi-omics, phenomics, and you can have information about the genome. Means when I say genome, so it is all levels starting from the chromosome till how it is translated or how a metabolite is formed. If you have that kind of information, that can be used for the knowledge-driven crop improvement. So now I am going very precisely to the omics approaches, which will be the focus of my talk, and I will be giving some examples that how we have been able to use those omics approaches to address some of such questions. So I have mentioned some of the omics technologies here. So not a single technology can answer completely any question. So it is better to integrate the output from different omics technologies and then reach to a conclusion or to identify certain the key target genes or the key target signatures which can be used for this purpose. So this is an overview of my lab, so what we all do. So we work on mainly rice and chickpea, chawal, chole. So it's a very important combination. So today also we ate, and any conference I have seen, so that is not there. Any meal is not there, in fact, without rice and chickpea. So we do all kinds of omics approaches, starting from the genomics, transcriptomics, epigenetics, non-coding RNA, regulomics, interactomics, metabolomics. The idea is to generate again different kinds of information, because every omics approach will give different kinds of information. And then if we can integrate all those information and can have the best candidate molecular signature. Again, I am every time emphasizing molecular signature because I am not talking about the genes only. The genes are one of the components, but it can be anything, a single nucleotide change or any kind of epigenetic feature. And if we know those, so these can be used for either breeding programs or like genome editing kind of technologies, so that we can have improved crops. Other than rice and chickpea, of course, I am working on few medicinal plants also. So, and that is basically in collaboration with other colleagues or other friends. So now, why legume? So I will be giving the example here, one of the story from this whole work which I have just shown you. So why legumes? Legumes, we all know it's very important. So they are highly nutritious. They have a lot of protein, carbohydrates, and they are good for soil fertility and actually for the sustainability also, so that you can have a good crop rotation and it can replenish your microbial diversity within the soil, so that you can grow next crop and you can have a good output. So it's important for the soil health, and also it gives the less carbon footprint. So on the left hand side, you can see that it takes very less water to produce dal as compared to any other kind of food. And chickpea, why? Because it's very important for India. India is the largest producer, but at the same time, probably we are not able to meet the demand fully. Some part now we have actually enhanced our productivity, but still some part we still need to import. And you might have seen two different types of chickpea, one the kala chana and white chana that we call as desi and kabuli type. So of course it's a very important crop for India. So this is a journey what we have done in the last almost 14 years for research in chickpea. I have compiled in one slide. So when we started, actually in the phase 1, so there were only around 30,000 ESCs corresponding to 5,000 genes were available, only 200 SSR markers. As I mentioned, any of the technology, if you want to implement for the crop improvement, you need to know about the DNA sequence or the DNA feature. So our first aim was to make it a enriched crop, means enriched in terms of the genomic resources. It was a kind of orphan crop for which almost nothing was known. So we started with the transcriptome sequence, we then went to the genome sequence, we generated some expression profiling maps, SNP maps. Then in the second phase, the trait-based analysis, means even more depth of the transcriptome analysis to know what are the genes or what are the features associated with this. Then epigenome analysis, high density SNP maps, expression atlas. And now we are going into the third phase, that where we are kind of integrating different data sets, employing multi-omics approaches. And recently we have started working on the metagenomics approaches. Of course, other than our group, there are many other groups throughout the country and as well as outside the country. They have contributed to make the chickpea as a now genome resource, genome rich crop rather than an orphan crop. So now I'll start with just two examples of our research. I will just quickly go through these slides, just produce and highlight. I will not discuss the data or results in detail. That I will be available after my talk. If someone is interested, we can discuss. So one example is the seed size diversity. So as I just mentioned, you go to the market, you see small size chickpea, large size chickpea, and you pay more for the large size. So it's an economically important trait. It is more nutritious also, and you pay more. So it is important for the farmers as well to have it the large size. So we wanted to know what are the molecular signatures associated with the small size and large size of the chickpea. So to start with, we did high throughput RNA sequencing data analysis. We took different stages of two different varieties of chickpeas. One was 13 grams for the 100 seeds, and other was 55 grams for the 100 seeds. At different stages, we almost generated around 3 billion sequence reads. And on the right hand side, you can see that the major difference comes at the S5 stage of the seed development, which is during where the grain filling is happening, the seed is increasing in size. And when we did in overall analysis, we found, this is the PCA analysis on the left hand side, we found that there are S3 stages, which is the late stage of the embryogenesis, and the S5 stage, where actually the grain filling is happening. So there is a significant difference in the genes which are being transcribed. So this was the first evidence that there is something happening at these two particular stages which are responsible for determination of their seed size. On the right hand side, we have shown the genes which are differentially expressed at different stages, what kind of functions they perform. So very interestingly, some of the boxes, if you can see there, we have highlighted, they are representing very important biological processes. So two of them I'll mention here. One is the cell cycle, and other is endoreduplication. Endoreduplication, cell cycle, I think everybody knows. Endoreduplication is that when your DNA replicates, but cell doesn't divide. So your fluidity level increases. So we found that the cell cycle related genes were very significantly enriched at the S3 stage when there is embryogenesis, and the endoreduplication related genes are very highly enriched at the grain filling stage, the S5 stage, where we found the major difference in the transcriptome. Then, of course, we did a lot of other analysis. So this is a kind of transcriptional regulatory network we generated, because we wanted to go more into the deep that how at these particular stages these genes are being regulated. So you can see the complex kind of structure. So where you will see that the orange or red colors are the transcription factors. Then there are cis regulatory elements, the promoters which have the conserved elements where these transcription factors bind, and then they control certain genes. So here again we had a kind of third line of evidence that the genes related to cell cycle and endoreduplication, they are being kind of regulated at S3 and S5 stage, which plays a role in the seed size determination. We then produced more line of evidences, because if you want to publish a good paper, so you just cannot just rely on one or two evidences. So we did some simulation kind of analysis based on the cell cycle gene. In nutshell, we have shown the result on the right hand side of the top. You can see that at S3 stage, most of the cells we found they were in the mitotic phase. And at the S5 phase, we found that most of the cells were in the endoreduplication, that is the G1 to S phase transition. So because at the G1 stage only the DNA replication happens. So by a simulation, we found that most of the cells, they were at those stages in the last series cultivar. Then we got some experimental evidence also in terms of the cell number, in terms of the flow cytometry analysis of the ploidy level, that again verified our observation. So now after transcriptomics approach, we did the genome sequencing also for different cultivars which are small sized and large sized. So here just it shows a summary of what all kind of modifications we found. We did a small or low level of association analysis. On the left hand side top, you can see that there were 6 genes we found that were very highly correlated with the seed size state. And we then further analyzed where those kind of SNPs are present, because when we compared all these, so we found there were certain SNPs which are present only in the small seeded variety. There are certain bases which are present only in the large seeded variety. And we found that when they are present on the right hand side, you can see if they are present within the protein coding sequence, they are basically affecting the protein stability. Again, this analysis is just computer-based, computational analysis. Now we want to study further. So actually, if there is a SNP, does it really affect the binding of the transcription factor? So basically, overall research is a mixture of the basic science and applied science. So a lot of questions we are asking at the basic level, that how this is happening, and then we are coming up. So that I will end my presentation with that. Some of the candidate signatures which are taken forward for the crop improvement purpose. So then the third approach we took is we analyze the DNA methylation profile. That whatever genes we found that their expression is being affected, at the transcriptome level, how they are being regulated. Again, in nutshell, so this is again a very high throughput single nucleotide resolution methylation analysis we did. And here we found that it was the methylation in the CG context. So again, I'm just coming back to in plants, the methylation occurs in three contexts, CG, CHT, and CHF. But it was the CG context methylation, and that too within the gene body, that we found was responsible for the higher expression of those genes into the large seeded cultivar. So we found that methylation plays a major role in the regulation of the expression of the genes which are associated with the seed size. So this is, we have just made consolidated kind of model that where in the top one, as you can see, the gene expression level, we saw that there is extended period of cell division and a higher level of endoreduplication in the large seeded cultivar. Then the methylation level is also higher, but only in the CG context and in the gene body. And there are certain SNPs which are associated, which are present either in the promoter region or within the coding sequence. They are responsible for this change in the gene expression. So in the meanwhile, we generated a very high throughput gene expression atlas also from 32 different organs of the chickpea. And again, we correlated this data with the known QTLs and SNP data set what were available in the databases. And we also generated our data for only few genotypes. And here are some of the candidate genes associated with different traits. You can see 100 seed weight, harvest index, shoot dry weight, plant height, root length density. Same way, the candidate genes associated with the stress response, the drought and salinity. So this is a kind of integrated approach we have taken to reach to some of the candidate genes. So this is a web resource also we have generated. So this is one example of the seed size trait in chickpea. Now we have few candidate genes and few candidate kind of signatures in the sense some SNP and the methylation epigenomic signatures also we have in hand. Some of these have been already taken forward by some of my collaborators to do the genome editing process. But for this, somehow we are not doing the kind of genome editing in our lab because chickpea protocol is not standardized with us. So they are taking forward this information to generate the plants or to modulate this seed size trait. So this is another example. I'll just quickly go through for the drought stress response in rice. So we have again used the same strategy here. We have different varieties available. We selected few of them which are known to be drought tolerant or drought sensitive. And then we did different omics approaches, transcriptomics, proteomics, metabolomics, methylome, whole genome sequencing. And even now we did for this, I'll have one slide for the chromatin confirmation as well. So we kind of again integrated these data sets. So on the left hand side, you will see that there are genes, there are proteins, there are metabolites, how they are regulated under drought stress. And while integrating all these three data sets, we found that it was a carbohydrate metabolism pathway, which actually plays a major role in regulating the drought stress response. And when we extended this work to more number of cultivars, initial analysis was just 2 rice varieties. One was tolerant and one was sensitive. But here we enhanced or we increased the number of varieties. So does it happen across multiple varieties? So that was the question. So when we did for 7 varieties, again we found the similar kind of output that there is a carbohydrate metabolism which is mainly associated with the drought stress response. Here also then we studied that how the genes are regulated. So we did methylation analysis. But here the methylation aspect or the regulation aspect via methylation, it was different. So we found that it was the CHH methylation, not the CG context methylation. And that CHH methylation in the promoter. And it was not a higher methylation, it was a lower methylation. That was associated with the drought stress tolerance. So what happens? So when there is a drought stress in a drought tolerant variety, so the methylation is removed. So because of the removal of the methylation, the expression of the genes which regulate drought stress response, that increases. And that leads to the drought stress tolerance. So then we went ahead further to see if this is regulated even at the higher level, at the chromatin conformation level. For this, we employed DNA-seq analysis. So we actually studied that where the nucleosomes are present in the DNA, like where the DNA is bound around the nucleosome and where it is not. So we call it the open chromatin conformation and the closed chromatin conformation. Closed means where there is a DNA bound around the nucleosome, and open when it is available for binding of the transcription factor or the gene regulator. So again, we compared two different cultivars under control and stress conditions. So this is all. I'm not going through the data set. I'm just telling you the output of all these studies. So here we found there were certain genes. Now we integrated all the three data sets: transcriptome data set, methylation data set, and even some histone modification data sets which were generated by some other group, and then our DNA-seq data set, which is chromatin conformation. We found that it is the chromatin conformation of the FIM gene, which along with the epigenetic modification, they control the gene expression. So it's the open chromatin. There is a larger number of the open chromatin into the drought tolerant cultivar, and that along with the methylation status is controlling the expression. So one of such gene, somehow we were working for the past 8 years in our lab. That was a candidate gene identified based on just expression profile, and we were trying to study its function, and we did transgenic arabidopsis and then transgenic rice. We found that when we overexpressed it, so the plant becomes more sensitive to the drought stress. But we could easily correlate now this data set with the omics technology. So this is again a kind of validation, whatever we are analyzing through the multi-omics approaches. So actually it is resulting into the candidate genes which work in terms of the functional validation also. Candidate gene based approach. So for this particular gene, we have even the CRISPR-Cas9, and when we are editing this gene, we are finding the stress tolerance of the rice plants is being enhanced. So this work is still in progress. We are in the process of analyzing this edited rice plants. So here I will stop just with this take-home message that integrated omics approaches, they can provide you promising target genes, not only the genes, the molecular signatures, and it is not only the nucleotide sequence, it could be the other features, most importantly the epigenetic features. They can also play in determining the phenotypic phenotype of a given crop plant or whatever phenotype you are interested in. So we need to look beyond the DNA sequence or just the expression profile. So now we are trying to employ even multi-dimensional approach, because in all these studies you might have seen one omics and overlapping with other omics or a third omics. So we are going step by step. So now we are trying to integrate all the data sets together in one go. If we can have such kind of bioinformatics tool, we are still struggling with this approach. So hopefully in next 2-3 years, we will come up with something that if we can have such a multi-dimensional approach where we can give the whole data set as an input, that will give more statistical power, and we can have better phenotype genotype association. Other things we are going ahead for the single cell approach. So as of now, we are employing the full tissue, like the whole root or the whole seedling. But now we want to study that what is happening at individual cell level, because even in a single root tip of 5 mm, you may have 20,000 cells, and every cell responds differently in terms of its environmental conditions. So now there are several methods available that you can do that kind of studies. So we are going into that direction. So with this, I would like to acknowledge my collaborators, lot of students who have contributed to this work over a period of last 15-20 years in my lab. Lot of funding obtained from the DBT, ESG. Earlier I was at NIPGR, so I used to get funding from NIPGR as well. And this has led to several publications in very good journals. Thank you very much for listening, and I will be happy to take any questions.
Thank you, sir. Any questions? Okay.
I can hear you. I can repeat your question.
DNA methylation में जो
क्वेश्चन करी है सर, वो क्या है वो 5, हाँ 5 NMC 6 MA, like what is this?
And currently we are talking about these numbers.
No. Next slide, please, sir. Next slide. Next. Last one slide. Last one. Yeah, yes. Epigenetic modification, DNA methylation 5, MC5, yeah.
So methylation happens at the 5 prime. So it's just a methylated C5. So it's nothing. There's the position. So where methylation is happening in the cytosine.
किसका position? Methylation का?
Yeah.
Okay. ठीक है. Thank you so much, sir.
You have the structure of the cytosine.
Yeah, I know.
The 5 prime and
3 prime. 5 to 3. Yes.
Is that either?
Okay. Thank you so much, sir. Any other question? Any other question? Thank you, sir.
Thank you very much.
I extend my warm welcome and invite our next speaker, Dr. Gyan Prakash Mishra Sir, Head, Seed Science and Technology, IERI, New Delhi. His major research areas are marker-assisted breeding of crop plants, breeding of lentil and mung crop. Sir has been awarded with several honors and awards as Plants Associate 2018, ISVS, Dr. Harbhajan Singh Memorial Award, inside membership, ICARDA Certificate of Recognition, ICAR Dr. Rajendra Prasad Award in 2020, IARI Dr. Ram Nath Singh Award, Biennial 2021, ISGPB Dr. Harbhajan Singh Memorial Award 2022, AICRP Rabi Pulses Best Center Award 2024. Thank you, sir, for joining us on the 8th AMIFOST 2025. Yes, sir. So which one? Which folder?
Very good afternoon, one more. Am I audible? Okay. So have you heard of this term, microgreens? Anybody? If yes, just raise your hand. If not, then it is always better that. Have you heard of this term like baby greens? No? Sprouts? Okay, done. So let's first understand what is the difference between, there are three terms. Microgreens, baby greens and sprouts. Baby greens are nothing, what we know in India as a saag. Okay, so there are three stages. First is your sprout, second is microgreen, and third is your baby green. So we'll be understanding why this microgreen and why this is coming as a novel food option for nutritional security. Sir was talking about something, he told there are two terms, food security and nutritional security. Any understanding? Food security, nutritional security? What is food security? Any idea? No idea? Okay. In 1960s, India was importing food. We are not having enough food. So we are importing from different countries. So that point in time, we were like food security was a major concern. After that, now after COVID, have you heard of this term antioxidant? Okay, iron and zinc are also showed. So now India is food secure. Now we are exporting food, but still issue is there with that nutritional security. There are many things in case of nutrition. So micronutrients are the major one. Iron and zinc, out of these are the two major ones. Plus different your vitamins are there. Okay. So this microgreen is nothing but this is for nutrition security, not for food security. Fine. Food means like we want lots of carbohydrates, proteins. The moment it comes to nutrition, it means that micronutrient, which is very much essential for our body. Right? So concern for health after this COVID is now more pronounced. We are more thinking about that better quality of nutrition, not only the quality of food, isn't it? So we'll be thinking about what is there in terms of nutrition, how much nutritionally rich my food is. So microgreens are what? Microgreens are they are tiny seedlings, okay, of around 3 to 8 centimeter height and 7 to 21 days. This is what is like definition of anything. So this is how it is internationally defined that this is how a microgreen is considered. If you are not giving light, it won't be considered as microgreen, it becomes sprout. If you are giving light beyond 21 days, it becomes baby green, which is also called as sod. Understand? So what is the requirement? Why will be going with this microgreen? So once it was studied, what was found that just because of this photosynthesis for 4 to 9 days, what we are going to give to these small tiny seedlings, it will be giving in general 4 to 9 times higher nutrition in general. But specifically, I'll be showing some details of this literature. It is sometimes even more than 40 times. Okay, so that is what it is. If you are comparing microgreens with its mature counterpart, so mature counterpart is what? Sag. Okay, so if I compare microgreen with that of baby green, so nutritionally microgreens are much better. Not only in terms of nutrition quantity, but also quality. Quality is defined by how it is being absorbed by our body. Okay, so bioavailability of nutrition is also better in case of microgreens if we compare with its mature counterpart. Fine. And then there are different nutraceutical effects. Okay, so for which microgreens are considered, and now it is like coming like a very big industry across the world, including India. Just you can Google and can find microgreens within 20 minutes anywhere in Delhi also. Right? Although as of now it is bit costly. So nutraceutical effect means reduced risk of cardiovascular diseases, protection against inflammation, oxidative stress, obesity, type 2 diabetes, and nowadays even cancer. Doctors are also now recommending microgreens to the patient of cancer. Right? And there are huge projections that it is going to increase. Somehow this table is coming, but still you can listen. Then there's nothing which is there on the lower part. So projection is that it is going to be huge, like in coming years, within 4 or 5 years, and still as of now, it is part of urban and peri-urban part. Okay, not under the rural condition. And one more advantage is that microgreens are always like organically produced. We don't have time, 7 days, just we are sowing and then harvesting. No need to apply any kind of fertilizer, nothing. Okay, so this is how there are 3 differences with this sprout, microgreen, and then baby green. So this screen is good, but this is not going to work. Anyways. So sprout, microgreen, baby green, basic difference. In case of sprout, no light at all. And in case of microgreen, 7 to 21 days, light is there. In case of baby green or baby leaf, it is going up to 40 days. Beyond that, it will be mature. And then no like agrochemical requirement, nothing is there. If you see the history of that, 1930 US, for the first time it was reported. What we write, that becomes history. If we don't write, it is not going to be considered as that we know the thing. Then 1970, 1990 for the first time in California, like different chefs were using it as a vegetable confetti. Confetti means that we are using it for garnishing purpose, different combination, different colors are there. And it is used only for giving color and texture to the any cooked food. For this purpose was started. And for the first time, 1998, the documented use of word microgreen was there in US. So from 1998 onward, this term started, we started using this term. And then there are different terms. Somebody is saying that NutriBoost, some other terms are there. But as of now, most accepted term is microgreens. So microgreens means we will be using multiple tiny seedling. So we are not saying microgreen. It will be like plural, microgreens. And 2000 North America, and as of now, 2024-25, biggest market US, followed by Canada and then Mexico. And the growth is very fast, and it is projected that 335 billion US dollar market is going to be there by 2029. Okay, that's a huge market. Global different types, it is classified: green type, farm type, end use type. And whatever I was saying, 2017, this market was only 25 billion US dollar, and 2024 it was $245 billion, and by 2029 it is projected to be 335. It means that there is a huge potential of this particular group of crop which is going to be there across the world. Why it is important? Nutritionally rich functional food. Functional food is different than any normal food. The moment this term functional food comes into the picture, it means that it is having nutraceutical effect. It is going to give protection against certain disease. Then rich source of vitamins, minerals, and secondary metabolites, that too in easily available form. Intense flavor, vivid color, tender texture, and then it is considered as specialty food, not like your normal food. Contains less nitrate, better bioavailable. What I am saying, if you are consuming, thing has to be available also to your body. So microgreens level, whatever is there, it is also very much available to the body, which is not there once it is getting mature, the same like plant. And then contaminant free, 7 days we are cutting, no chemical is used, and then ease in production and handling. And generally for any adult, it is 5 to 10 grams per person per day. Okay. So functional food, we understand. Space food, always, this is a part of space mission. Always it is being grown, and then for nutritional security it is used, and literatures are there. Skin care formulation. The sun protection formula, SPF 20, 30, whatever we are using. So basically that comes from microgreens only, from different microgreens, edible, non-edible, so that antioxidants are isolated, and this, because this is going to give cleansing and then exfoliating effect to the skin. Then for nutrition, the security of harsh areas, Ladakh, then different areas where it is very difficult. Sometimes there is no aircraft at all, no fresh food. There also it is being grown, and even DRDO Army is going will be growing under very high altitude condition where fresh food availability is not that much. First paper came in 2013 by Sao et al. And it was from University of Maryland, where for the first time they studied 26 microgreen. And what they found that it was quote unquote microgreens were 4 to 40-fold more concentrated in nutrients than their mature counterpart. Now we understand microgreen and mature counterpart. We never compare it with the seed and anything else. And there are few examples I'm not going to detail, like amaranth 1.14, red cabbage 0.04 microgram per gram of fresh weight. If you compare with the microgreen, same, it becomes 4.09, 2.77. Hundreds of papers are there which says that how much things are more. And what we compare, like vitamin K, vitamin C, provitamin A, these are all vitamins, and still they are a type of your antioxidant. All the vitamins are not antioxidant, they are part of antioxidant. Then carotenoids are there, vitamin E is again very high. And then wherever we see this colored, like purple cabbage, red cabbage, then different type of colored vegetables are coming. So the moment color is there, it means that it will be better than your green or white ones. Fine. Have you seen like red okra? Anybody? Okra, lady finger, it is available in the market, and it is much better than your normal green okra. Different varieties are there. So lots of paper, I just wanted to compare a few. Even the research paper says that minerals are also very high. Okay, although we are using neutral media, but still, if we will be using some media where nutrition is better, so what was observed that these microgreens are having more of a nutrition in terms of your even iron, zinc, potassium, everything is high in case of microgreens. Fine. Then I'm not going into detail. Cardiovascular diseases, number of examples were there. For example, red cabbage, altered lipid and cholesterol level, polyphenols, and then anti-diabetic, anti-obesity, anti-cancerous. Number of microgreens are reported. Amongst that, broccoli is the number one. If you ask which microgreen is at one, across the world, it is broccoli. Okay, broccoli otherwise is also considered better than any other one. And microgreen of broccoli is again the best amongst whatever microgreens are there in terms of nutrition, and also across world. The microgreen which is maximum produced, sold, consumed is broccoli. Fine. So, and this falls under the broad group of which is called as brassica. Brassica is a big family under which this broccoli comes, and many things are coming. Okay, so this lowers the ascorbic acid and then less response against cancer. Lots of studies were there for different cancer line. It was found that broccoli microgreens are best. Right? Then anti-inflammatory activity, number of microgreens were there, and then number of studies were there, and again broccoli and red cabbage were considered as best among all. This is one comparison, just 25 grams were there. Once we purchase, we generally find that for any family, for any day, 25 gram is enough, even if it is a family of 4. So it was considered that 2.5 gram seed is going to be microgreen, 25 gram. In case of your brassica, which is nothing but then 25 grams and then mature greens and then percent daily value. So here also what we found that most of the nutritional component of microgreens are better than even seeds or mature greens. Breeding strategies are now also being applied because any seed is not good. We can breed for microgreens, higher yield, and then as of now if you see across world, it is going with this color. Different colors are there. So breeding for color is also going on, that which color is better than anything else. Conventional approaches are there, biotechnological, molecular approaches. We'll be screening large number of genotype at like PUSA, and then we need to find which one is better than anything else. Right? So molecular approaches, transgenic approaches, even genome editing, because we want in 7 days more nutrition. So we'll be targeting certain genes so that it is going to express more in that 7 days rather than we'll be waiting for that to grow. And hybrids, we don't want hybrids in case of microgreen because hybrids are very, very costly. We would just want a microgreen which is growing very fast. So for that, there is a one term which is called as ESV, early seedling vigor, in what we say in case of seed science and technology. So early seedling vigor means 7 days. It has to be much faster than the same variety which is growing other variety which is may not be that fast, isn't it? So now that research is changing very fast. So light, different group of lights for there, like red is there, different nanometers are written. So red, blue, and then combination of red, blue, and yellow. We will be trying. So my PhD student, she is also working on that. So what we found that, and number of papers are coming, the moment we change the light, lambda, so nutritionally also same varieties behaving differently. So we are now optimizing which light combination is going to be better. So sometimes red is increasing, sometimes red is not increasing. So we need to find at what lambda and how much duration it is going to be better. And then we'll be using that combination for having more nutrition with same line. Right? So different combinations for their mustard, beet, parsley, lettuce. We have developed one kit at IRI PUSA. So this is very well taken by different schools. Children can, if you wish to have, you can use it. So it will be given 3 different combinations, and always what we make sure that we should include a radish, because whatever microgreens one grow, any person who is new in case of microgreen, I always suggest that go ahead with radish seed, and then it will be best to grow. Very easy to grow. If you can grow radish, then you can then play with other microgreens, which is not that easy to grow. Okay, so we'll be adding this, and then school children, they are more than happy to, and then teachers are also happy. They themselves are growing, and then they are also consuming, because it takes only 7 days. And then we'll be giving 3 seeds, so the same tree can be used thrice. And then different forum, I try to like popularize this also. We are working like in case of, if you happen to visit IRI PUSA, must visit. If you have not visited, try to come, find time and come to PUSA and see how we are working. And then we used to have like Kisan Melas over there, and different kinds of fairs are organized. There also we like push this microgreen kits over there. How to grow, soil or water, both are okay. Okay, different airlines, they prefer to grow under water condition, which is also called as hydroponics. But in otherwise, across India, across world, what we prefer in soil or soilless medium, what we call. Okay, so cocoa peat, coconut peat, that powder is there, that is called cocoa peat. So cocoa peat is the best medium to grow. But bigger airlines, they are having their own system. They always go with the like a hydroponic system for the production of microgreen. Just broad understanding, if hydroponic system, like sticking of any kind of soil particle to the microgreen, very clean microgreen, we can have all control. If we'll be using any kind of like coco peat or any kind of soil media, so little bit that stickiness is there and we need to clean it in a more precise way. And hydroponic is always costlier if at the initial cost is high, otherwise later it becomes fine. Different kind of systems were there. It is all available on like one can depending on the need, one can go ahead with like nutrient film technique is there, aeroponics, vertical tower system you must have seen, then wick system is there. Everything is available in the market depending on need. What is the size, what is the requirement, based on that we'll be selecting. It's not like that we'll be selecting all. Then drip system, ebb and flow, microgreen trays, out of different system. Like in case of household system, even at the commercial level, Delhi or any Mumbai, Bangalore, microgreen trays and countertop microgreen tick, these two last one are mostly being used for microgreen production. And then hydroponic system, as I said, very simple systems are also available. Bigger system are also available. It all depends what kind of need is there. Accordingly, we'll be going with that. But whenever we prefer to grow microgreens under open field condition, because we need to have control system for having assured nutrition about that. We want to have assured nutrition. So for that, a control system is required. And then hydroponic is one option. Otherwise, also we'll be going with a tray kind of a system. Proper lighting is there. And then in case of research, we'll be changing different light combinations and we're trying to find which combination is best where nutrition and yield both is high. Now, how we can shift? Because shifting our food habit is very, very difficult. Easiest way, first start using some small tray and which is always there at home. And then seasonal seeds available in the kitchen like mustard, mung bean, fenugreek, coriander, many seeds are there which can be easily used. Do you know the difference between seed and like grain? Any idea? What is seed, what is grain, or both are same? Same or different? What we consume, that is grain. What is being sold in the market in the packets, that is seed. Seed is a legal entity. That's the bigger, like at least this much understanding is required. Once we say that it is seed, we know that how much germination percentage, what are the things are there. But the grains which is available at my home or your home, that is also equally good. It will be germinating. So one can start with that and then customize based on whether you are from South India, North India, what kind of food habit. It can easily be customized and included in that. Only thing is that we are supposed not to boil it. 80 degree up to that, that's fine. When we did one, like 2-3 times we worked with IHM, that is there at the Pusa Institute of Hotel Management. They prepare different kind of dishes. Like it's not like that we can add any amount. So how much can be added per 100 gram, then 250 grams. Accordingly, they are optimizing. So lentil microgreen, then red radish, then green radish, then mung bean lentil combination, chila chutney, different combinations were there. So we got some data that how much can be mixed and which is acceptable to Indian condition and Indian taste. So based on that, different research is going on. And omelette, noodle, pasta, pizza, easiest to grow. Anybody can grow, children can grow, you can grow, and then you can straight away use it in your food. Only thing is that this slide I got from them only item. So baking, boiling, stir fry, saute, steaming is allowed. Try to keep it less than 80 degree. Nutritionally, it is going to be same. If you are crossing more than 80, it is not recommended to be used. One different combination, I was having one project from DRDO. So we were working on different groups of microgreen under Ladakh condition and Delhi condition. Ladakh, if you have visited, it's okay. Otherwise, just understand that temperature is low, ultraviolet B is very high, and duration of again, it is very high. So based on that, there is differences in that nutrition, and it is mainly because of the stress condition. Ladakh is more stressed and Delhi is not that stressed. So what we found that different combination, different genotype, there is a variation, huge variation. So we could identify one genotype which is best performing under Ladakh condition in case of lentils and mung beans. These are just data, not required. Then microbial contamination is very much known in case of sprouts. So, but in case of microgreens, since we are giving light, we are giving like proper aeration. So that was not at all reported. And whatever reports were there, you can see in the third one, like inoculated under hydroponics, contaminated water into irrigation. So we intentionally contaminate and then we see what is that level of contamination. So we also try to study different kind of contamination, but it is never ever found that any kind of contamination of microgreens were not there. So vast on vast different combinations we tried. So fungi, aerobic bacteria, Shigella, E. coli, these are the major one, which is like part of human contaminants. So we could not find, and whatever is there, it is less than the internationally accepted standard. Then RNA-seq was also done. We try to find like why there is a difference and why microgreens are better under Ladakh condition over at Delhi condition. So we did like different stages, and then still we are trying to like analyze what are the major genes combinations which is responsible for that genes are there, why it is just because of change of in temperature environment microgreens becomes better or poorer. So as I was saying, biotechnological ways were there and different ways. So out of that, even gene editing is going to come because transgenics are not at all accepted by Indian government, although many countries are accepting. So SDN 1 and 2 can be like applied. Even for microgreens, we'll be targeting a few major genes for antioxidant, and it will be times more than what we are getting right now. So once we wanted to grow microgreens, one has to be a bit careful that we are supposed to use untreated, uncoated. In case of seeds, mostly it is treated and coated. We are not supposed to take that kind of seeds, and then no need to go for very highly priced seed like hybrid seed, and avoid deep or shallow sowing of seed. That is also essential. But once we start doing it, when you can understand, and we should not supposed to like give excessive irrigation, using normal soil, and must add lot of organic matter and grow them in a sunny place, or always try to grow where proper lighting is there, and harvesting at right stage is very much essential. Number of entrepreneurs are there. I'm in touch with them, mostly whosoever is working on microgreen. So Vidyadharan from Chennai, Kia Sharat from Mumbai, Krishna Reddy, Shamila Reddy, then from Hyderabad, and then Babu Paul, he is the founder of GNH Botanics from Cochin, Mayank Chaturvedi, he's from Delhi only, and Vikram Udayagiri, they all are having their own websites where they are working for multiple things, including microgreens. So this Babu Paul, he is a designer. He designs different, and then he gets the patent for how to grow microgreens in a smaller pot, a bigger scale, then sprouts and all those things. He's not doing microgreens business, but he's designing and then patenting and then selling his designs to different companies. I also try to give different, like in case of DD Kisan or wherever I find, Mayank Chaturvedi and his wife Sarika also did. And then we try to publish whatever is there. And from like in India, we are the first to publish as a research paper in case of microgreen. And then we'll be still in the process to find what are those things, and then different kinds of like literature, whether it is Hindi and English, we try to do it. So what is the take-home message? Broadly, more practical way to add fresh and nutritious produce to your meal, and can be added in a number of ways. It all depends what kind of local cuisine we wanted to use, and cultivation can be promoted at home at kit, and then smaller these kits are very much easier to be used and need more research is required. An institution like IRI and many even from MIT also, we can work on more like deeper research is required rather than just cultivating and consuming this microgreen. And retail price, whatever calculation across world says, approximately 5 to 11 times is the minimum. It's not a mean. Is higher than the cultivation price for depending on what kind of seeds we are choosing. And then entrepreneurship development through institutions like IRI and other is going to add more science to the microgreen. So I would like to acknowledge the funds received for this project like DRDO, ICR, then Director DIHAR. DIHAR is the Institute Defense of Altitude Research at Leh, or then Dr. Shari Praveen Brahma Singh, all those who are like associated with this particular project. At last, I would like to end the presentation with saying, let your food be your medicine and medicine be your food. Thank you. Thank you. Thanks a lot. If you have any question, I'll be more than happy to answer. Yeah. You're very close. I can hear.
Hello. Sir? So, sir, you mentioned about incorporating the microgreens in a daily diet, but it's not very famous. So, sir, how can you like suggest some methods if we can incorporate microgreens in our daily lives without compromising the nutrition value?
The moment you are going to add microgreens in your daily diet, it's an addition. So you are going to have more of a nutrition, and easiest way first to grow is it is easy to grow because there are a number of seeds which are easily available at home. So we can first start growing, and then it's just like a replacement of garnishing. Okay. So whatever we are consuming, in just like coriander, you can first think of replacing that coriander with this, but not 100%. what was generally accepted in case of India, 25% of microgreen and rest of coriander or mint is going to work very well. You will be having very different tastes. And then otherwise also, if you go to any 5-star and 7-star, microgreens are always added as a garnishing purpose. So this is part of our food system, but not regular at our home. That's the only thing. As I was saying, pasta, pizza, you can always add. So different things we are adding, we are not knowing what we are actually adding, but still we are adding. Okay. So we are in the process of even making microgreen tea. So dried microgreens, again, very intense in case of nutrition. So that much intensity is not accepted to our tongue. So what we do, we will be adding part of that microgreen plus some other which is accepted in case of India. So based on that, we'll be doing. But idea is that nutrition has to be better. So again, you are not supposed to eat like 25 grams. 5 to 10 gram is like enough to find DV. DV is your dietary value per day. Okay. And brassicas are very accepted for Indian ones. Okay. So brassica means cabbage, cauliflower. Okay. So broccoli, these are forms under the brassica. So otherwise also we'll be using like sarson ka saag. This is brassica only. So this microgreen of brassica is much better than at the saag level. But saag is also required. So cooking is not permitted. Don't go beyond like 80 degrees. Fresh is always better to be used. I have not included like storage other things. Otherwise, we can keep this at 4 degrees. Lower part of your fridge is 4 degrees. Upper part is minus 20. So 4 degrees, 4 days easily can be kept. Most of the brassicas. Fine.
Thank you,
My
sir.
position go with brassica first, then adventure with anything else.
Thank you, sir.
Good afternoon, sir. Good afternoon. So last year when I was in MasterChef, so we used a lot of microgreens in the audition. But afterwards, I tried to find these microgreens in the local market and it was not readily available. So right now we are planning for a startup to provide the nutritionist food in the market. And we were in search of these microgreens, but again, it was not easily available in the market. No. So, yeah. Continue. So, if it is that much nutritious, so what is lacking in the market that they are not in the major markets? There are 2-3 things. What you want? You want fresh microgreens in the market, or you want to get the seeds for the microgreens? Fresh microgreens. Okay. See, what I could understand, the cost. Cost is very high. You can get microgreen within 10 minutes here also. But what is the cost going on as of now across India? 25 grams, 200 rupees minimum, including the transportation cost. That is first. And it's still microgreens are consumed at 5 and 7 stars only. And wherever else you are going to see microgreen, it has to be like someone who is nutritionally very cautious. They are using microgreen. Otherwise, microgreens are being used where doctors are recommending that you consume. So that clarity, see, your wheat grass is nothing but microgreens only. Yeah. Okay. So once Baba Ramdev says that wheat grass, even powder form, everybody is using. We are not knowing what is the concentration, anything else. So somehow I feel that in India, nutritional understanding and whatever microgreens are sold, nobody is writing what is the nutrition content. So these are the things, and somewhere lack of knowledge is the major one. Otherwise, there is one thing called NSC, National Seeds Corporation. They are also now selling microgreens kit and microgreen seeds online also. So availability of seed, availability of kits are there, availability of knowledge is there, books are coming. So from India, I'm saying. Abroad, it is way back, it is running from 1990. So your answer is that it is the price as of now and that understanding. Unless until market is going to grow, there won't be any reduction in the price. But if somebody wanted to grow it, you wanted to have seeds, so NSC is there, PUSA is there, you'll be getting any kind of seed. Fine. There are certain entrepreneurs, if you wanted to be in touch, there also you can get. Sure, sir, sure. So it is affordable to grow at home instead of affordable. Once you need to come and then we can sit for half an hour, then I can tell you what is the issue. Issue is it has to be consumed in 7 days. So if you are knowing 7 days before that which chef, which person wants what. Otherwise, you cannot keep it for a longer period. Nobody wants that it should be kept under the freezer and then it comes to my home. So it comes always fresh. Cut in the morning, So whosoever is in this microgreen business, they know around 7 days before that, I am going to deliver this much fresh microgreen to this particular restaurant, hotel. Fine. And this is a way fresh microgreen. Otherwise, microgreen kits are there in the market. Online, you order it, you will be getting. But they are not telling the seed. That is the biggest problem. Once you get them, you find that these are the seeds which is not at all required or may not be of your choice. Thank you, sir. Thank you for that. Thanks a lot. At least you are knowing this much. At least I'm more than happy. Yeah. Anybody else? Thank you. Thank you.
Thank you, sir. I will just. Oh. Thank you, sir. I call upon our next speaker, Dr. Jitendra Kumar Vaishya, Research Officer and R&D Head, Ministry of Ayush, Government of India. Sir has several publications in various journals of repute in national and international. Sir is also a fellow of National Academy of Sciences, Prayagraj.
Good afternoon to all of you. Before we start to my talk, I would like to thanks to the organizer for inviting me for today's event to deliver lecture about the importance of medicinal plants, especially in respect of nutritional value, and how the Ministry of Ayush is working on it, and how we can include medicinal plants in day-to-day life. So, due to some unavoidable circumstances, I am not showing you PPT. I will brief about the activities of the Ministry of Ayush, Government of India. National Medicinal Plant Board, Ministry of Ayush का एक section है. One section who is dealing the medicinal plant related issues in the country as well as abroad. Like we are today discussing about that this, like in food, how we can use as a supplement, how we can include the herbs. So National Medicinal Plant Board having the central sector scheme under which there is provision to apply project like grant writing. Under that you can that faculty members can apply project for getting the approval of establishment of herbal gardens. For the research and development, we have the separate component for the R&D under which find out the novel compound, how to develop the value added products. जैसे हम यहाँ पे अभी कुछ खाने पीने की चीजों की भी चर्चा हुई कि कैसे हम प्रिपेयर करेंगे, कैसे हमारा जो सीरियल क्रॉप है उसमें कैसे हम आगे बढ़ सकते हैं. तो द सेम थिंग्स विद द मेडिसिनल प्लांट्स लाइक ग्रीन टी. अभी आप मार्केट में देखते होंगे तो ग्रीन टी में आपको हिबिस्कस का फ्लेवर है, हिबिस्कस, कैमोमाइल, tulsi. So like this, some of the herbs is very important, very much important that can be used in day-to-day life. We have the so many components like R&D, then IEC activity under that. We also promoting the capacity building trainings for the farmers. Then we know that SDG, Sustainable Development Goal, SDG 3, good health, well-being. Under that also our ministry is promoting medicinal plants sector for availability of the raw material, genuine raw material in the country for the Ayush industries. And Poshan Vatika mandate recently added under tribal ministry, under which we have identified some of the medicinal plants. Those can be used as a nutrient We know that micronutrients, like I can give one example, moringa, drumstick, that fruit also use edible, then leaf का देखिए कैप्सूल भी आता है, पाउडर भी आता है, then that is good source of nutrient, माल न्यूट्रिशन में भी देते हैं बच्चों को. Then for memory we can use Brahmi will act as a memory enhancer. Then गले में कुछ खराश है, अडूसा, आधा डोडा वेसी का, like this. तुलसी, जिंजर, हमारा जो किचन है, किचन में भी बहुत सारे मेडिसिनल प्लांट्स स्पाइसेस के रूप में यूज होते हैं, जैसे हम लौंग यूज करते हैं, देन दालचीनी, अजवाइन. So like this, whatever the spices available in our kitchen, that is also used as a home remedy. I am talking about the prevention, not cure. So medicinal plant is good for the prevention of any disease. Like COVID, before COVID, our ministry has promoted, National Medicinal Plant Board has promoted Amruta for Life Giloy campaign, boost immunity. And after COVID, we have launched Ashwagandha campaign as a health promoter. Ashwagandha is reduce the stress. So many property under and in the Ashwagandha, anti-aging property also. Now, right now, we this last month, our Honorable Minister of Ayush has inaugurated and launched Satauri campaign. That is also good for the women health. Satauri can be used for the memory, then especially for women also. And Satauri is known as Narayan. 80 type of neurological disorder, जो भी हम महानारायण ऑयल मार्केट में मिलता है आयुर्वेद का, and that also the Satauri is there. Satauri अफ्रोडाइसिक भी है, and Satauri से फार्मर का इनकम भी बढ़ सकते हैं. A farmer can get a root as well as seed also. The same things with the Ashwagandha. Ashwagandha plant, जब हम हार्वेस्ट करते हैं, that root also farmers can get the root as well as seed. Both they can sell and they can earn more money. So like this, our ministry is working in the field of medicinal plant. We also making the policy. Like I discussed about the tribal ministry, and Ministry of Ayush has signed so many MOUs with the other ministry as well as the other country. Last year, myself visited Vietnam as a part of delegation of Ayush, and we have a discussion there also how to promote and do the promotion of medicinal plants jointly with India as well as Vietnam. So we have research and development component under which we promote a good agro techniques to develop the good agro techniques. Then how to find out the novel compound. Then under that our ministry will provide the manpower also, like JRF, SRF, for the execution of the work. Then develop the standard protocol. Like कोई भी एक procedure developed है, on that also if you can change the procedure and develop new protocol, finding out the substitute. जैसे कि 10 मूला का मैं एक एग्जांपल दूंगा. 10 मूल में 10 drugs होता है. That root is used for the making the drug. If we can pick the root, plant will die. So we can find out the other substitute with R&D, like whether the same phytochemical constituents is available in young shoot or leaf or flower or fruit. That such kind of projects NMPV will grant to the good organization. And I also feel happy to share with you all that under Ashwagandha campaign, our ministry has supported one project to Amity University, and Dr. Swati Madan is the PI. So here also we have given the grant, and they will promote and they will develop the one brochure, and by which they will also make aware to the students and the local people, farmers. And beside this, we have the scope for establishment of nursery, And like this food technology department, we have the value added product. वैल्यू एडिशन के भी प्रोडक्ट नेशनल मेडिसिनल प्लांट बोर्ड सपोर्ट करते हैं, लाइक कुकीज, चिंगम, जैसे चिंगम है, चिंगम में अगर हम गिलोय का एक्सट्रैक्ट डाल दें, दैट विल बी गुड फॉर द किड्स विथ प्रॉपर एसओपी, दैट विल हेल्प टू बूस्ट द इम्यूनिटी. Like this work, like papad kha rahe, juice pee rahe, on that we can add some supplements of medicinal plants products. So such kind of work National Medicinal Plant Board will support. So and specific campaign we are already promoting right now, and we have the our website for market linkage, e-Charak app, e-Charak. So anybody can download it and they can find out the raw material price and who is the buyer, who is the seller. Direct platform we have developed. We have our website, so whatever the left in my lecture, that you can get in our website. If you have any question, you can feel free to contact us anytime, so we can provide the that answer. So once again, I would like to express my sincere thanks to the organizer, especially head of the department of the food technology, then all the faculty and my senior also. And thank you all for listening me. Once again, I would like to thank my competent authority, Chief Executive Officer, Dr. Mahesh Kumar Dadhish sir, who has given me permission to participate today's event and deliver talk about our scheme. So next time, if I will get time, I will get chance, definitely I will elaborate in details. Due to some unavoidable circumstances, I am leaving with a short time. So sorry for that. I'm expressing my apology. Thank you so much for listening me once again. Thanks to the sir and madam and my seniors are also for inviting me and giving me opportunity for today's event. Once again, thank you. Thank you all. Jai Hind, Jai Bharat.
Thank you, sir. Thank you for being so impromptu in your speech and on a last minute. Thank you, sir. I request HY sir and Dugesh sir to kindly come on stage to felicitate our speakers. Sir, just wait for the presentations going. I request HY sir and Dugesh sir to kindly felicitate Jain sir with a memento. I request HY sir and Durgesh sir and ma'am to kindly felicitate Gyan Prakash sir with a memento. Request HY sir, Durgesh sir and ma'am to kindly felicitate Jitendra sir with a memento. Durgesh sir, I request you to please join us on stage. Thank you, sir. Thank you, ma'am. Thank you, sir. I move towards with the technical session two, nutrition, health, and functional foods. I request our session chair and our speakers, Dr. Sarkar and Dr. Sriram Yadav sir, to kindly join us on stage. I request our session chair, Dr. Minju Niraman, to kindly join us on stage. I welcome the session chair, Dr. Nirja Hajola, for Science and Regulatory Affairs, Yakul Denon, India Private Limited, Delhi, India. More than 20 years of experience in developing, planning, and implementing scientific programs and appraising scientific information and adapted written and oral communication to a range of audience, including health professionals and consumers. Led and felicitated a multidisciplinary team with a wider reach of scientific knowledge, organized international and national symposia for scientific deliberation on the emerging science of gut microbiota and probiotics. She is also responsible for regulatory compliances, including government approval processes, ensuring compliances with industry regulation, necessitating scientific data for approvals, and validating information and packaging and labels. Fostering and maintaining strong links with key opinion leaders, both at national and global level. She also possesses strong communication skills with a deep commitment towards professional excellence. She can handle multi-priorities with abundance of action. I request ma'am to kindly join us on this and please share your word of wisdom. It's okay, ma'am, if you want, we have the mic also. Like that's fine. So, a very good evening. And before I start, I would like to thank Dr. V.K. Modi, Dr. Monica, and the entire team for this invitation, for having me here. And I think functional foods is the need of the hour. So it's very timely and I think a very, very apt topic for me to chair. So just to give you a background about the session, we all know that malnutrition is one of the biggest developmental challenges facing the world. You'll be surprised to know that 805 million people still go hungry every day. Over 2 billion people lack essential vitamins and nutrients which are needed to grow and live healthy lives. Primarily, this is because of diets which are rich in starch and provide calories but not nourishment. At the same time, we have 1.4 billion people worldwide who are overweight or obese. Now, in India, we are battling with the triple burden of malnutrition. We have undernutrition at one end, we have overnutrition, and we have hidden hunger. So, the double burden of malnutrition is prevalent with 3.5 billion people, which means that 1 in every 2 people on the planet is malnourished in one form or the other. In India alone, 185 million people don't get enough nutrients. Hidden hunger is especially prevalent among children, and you'll be surprised to know that more than 70% of India's children under 5 years of age are deficient in vitamin D. More than 50% of children in the country lack adequate levels of vitamin A. And these deficiencies have contributed to high levels of stunting, wasting, and an increase in the number of underweight children. So, affordable nutritional supplements taken in the form of capsules or tablets can mitigate the symptoms of hidden hunger, but can cause toxicity if consumed in large amounts. And therefore, today we are in the era of functional foods, which are a low-cost approach to curb micronutrient deficiencies. So, what really are functional foods? They are foods which give you nutrition and health benefits. And a very, very simple example of how functional foods have helped our country is the example of iodized salt, which helped to reduce goiter significantly in the country. Now, the concept of functional foods emerged in or originated in Japan in the 1980s. And this was when they did not have enough medicines, and that is when the government agencies started approving foods which had proven benefits in an effort to better the health of the general population. Today, Japan has what is known as the FOSHU label, that is the Food for Specified Health Users label. And this label is given to products which is validated for their health benefits, besides giving you nutrition. Now, in 2016, the Food Safety and Standards Authority of India released standards for the fortification of 5 staples: rice, wheat, soy, salt, oil, and milk. And today we have regulations in place to fortify milk with vitamin A and vitamin D. In recent years, why have functional foods become so important? And this is largely because of an increasing health consciousness, the growing concern about nutritional deficiencies like hidden hunger, and the growing burden of antimicrobial resistance. It is estimated that 10 million people will die due to antimicrobial resistance by 2050 unless and until an immediate action is not taken. And therefore, there is an immediate need for interventions in the form of functional foods or nutraceuticals, which can limit the need for antibiotics, and in some way, functional foods and nutraceuticals today, which give you health besides basic nutrition, seem to fit the bill. So many of these functional foods are associated with potential health benefits. They may prevent nutrient deficiency, they may protect against diseases, and therefore you have foods which are general foods. It could be in the form of your fermented milk, it could be in the form of bread, it could be in the form of juice, which could be rich in antioxidants. And these antioxidants help in neutralizing harmful compounds, which we all know as free radicals, to prevent cell damage and chronic conditions, including heart disease, cancer, and diabetes. So functional foods, being a food, could actually reduce risk of these non-communicable diseases. Some functional foods are high in omega-3 fatty acids, a healthy fat which is shown to reduce inflammation, boost brain function, and promote heart health. Others are rich in probiotics, prebiotics, which help in improving gut health. So today we have a range of products which can actually improve gut health, because when we're talking about nutrition, it goes hand in hand with your gut. So if you have a healthy gut, whatever food you're consuming, whatever food you're eating is going to be digested properly, the nutrients are well absorbed, and you get all the energy that is required for growth and development. So it's not surprising that the Indian functional food market is estimated at 12.28 million US dollars in 2023 and is projected to reach US 27.9 million US dollars by 2030. And this is growing at a CAGR of 14.74%. So dairy products hold the largest share, and this is followed by bakery and cereals, and there's an increasing demand for convenient and healthy snacks. So with that, I have set the stage for the session, and I'm going to invite our first speaker to please come on the dais and give us a talk.
ये पीपीटी है इसमें
कॉपी नहीं शेयर करना है।
ये कॉपी नहीं करना ना डायरेक्ट
फुल स्क्रीन में कर दो, फुल स्क्रीन में कर दो।
I humbly invite our speaker, Professor Debavrata Sarkar, Professor Biosciences and Bioengineering Department, Indian Institute of Technology, Roorkee. In 2010, he graduated from Indian Institute of Technology, Kharagpur, with a PhD in plant natural product biotechnology. His postdoctoral research training was in mass spectrometry at Technical University Braunschweig, Germany. A major focus in his research is the development of artificial intelligence with biosensors for early detection of plant microbe interaction in economically important crops planted at an asymptomatic stage using mass spectrometry based metabolomics. In addition, he has been awarded UST's Young Scientist Badge, the IIT Roorkee's Outstanding Research Award. Additionally, he is a visiting professor at Institute of Plant Biology, Technical University, Germany. As of today, his group has trained 14 PhD and 8 postdoctoral fellows. He has published more than 100 articles in prestigious journals and holds multiple biosensor patents. He serves as a member of several scientific bodies and as a member of several editorial boards of India. Thank you, sir. And today, sir, would be speaking on the future of farming, AI-driven sensor-based plant disease detection and quality monitoring. Thank you, sir, for joining us today.
Thank you very much. Introductions. It was, of course, not brief, and thank you very much. And before I start, first I will thank Dr. Monica, Dr. Durgesh, and Dr. Renu for inviting me. And as you say that I belongs to the Indian Institute of Technology, so throughout my talk, you can see the flavor of technology that orchestrating the science. So this topic of this conference is food security. The food security is a very broad domain, and you can find our contributions is very little. So what is our contributions and what is the journey in this last 14 years that you will see. So we will traditionally talk about the food security. So firstly, it deals with the enhancement of the yield. Then enhanced and customized nutrition. So just before my talk, Professor Mukesh Jain has given a wonderful outlook of how to enhance the yield as well as the nutritional profile. And the third parameter is very crucial. This is reversed to how to minimize the loss which is due to the biotic and abiotic stress. So what my research group is focusing in the third parameter, that early predictions of the plant diseases utilizing AI-based sensors. So my research expertise, so I am a mass spectrometry guy, and then my research group developed expertise into the development of the biosensors. So this is what is called what is conventional agriculture and what is the precision agriculture. In conventional agriculture, what happens after the pathogen infects the plant, there is a symptom. And then once the symptom appears, then farmers can go look into the symptoms, and based on the disease detection they have, then they will go for the next level of control measure. But in most cases, that is too late because already the propagules are there and it infects the entire field. What is our goal? That if we can develop a sensor which can predict the disease at an early stage when absolutely there is no symptom. So we will take an example of some diseases of the rice, which is bacterial blight, blast, as well as the sheath blight disease. So what is the current practice of the management of the rice diseases? Either to develop better genetic resources through the genome engineering and other transgenic approach, which is of course very good, but always takes a time. And whenever most of the cases, when you develop the resistance into the plants over the year, by the time pathogen also develops a new level of virulence. So what is the concept is, this is a rice leaf. The first one is the healthy leaf, so absolutely there is no symptom. In second case, infection is already there, but there is no symptom. And third case, the leaves are infected and symptoms are there. What is our target? Our target is to detect the disease into the stage 2 when absolutely there is no symptoms. So normally what happens, when this pathogen infects the plant cells, pathogen will enter inside the plant cell and then it will release some of the effector molecules. And those effector molecules will trigger some of the metabolic reprogramming, resulting into the release of some biogenic volatiles. And what is our interest to identify those biogenic volatiles, and if those volatiles can be used as a target for the early predictions. So this is our concept, that there's a plant cell, the plant cell infected with a fungal or the bacterial pathogens, there would be a plant microbe interactions. And when there's a plant microbe interaction, it will cause a metabolic reprogramming, and that metabolic reprogramming will lead to the synthesis of novel volatile biomarkers, which could be used as a target for the sensing. So this is what we speculate when we started this research in 2014, almost 11 years back. And then I will show you at which place we are. So when there is a pathogen challenge with the plant will emit certain volatile. But unfortunately, these volatiles we neither can see or neither can sense. So we have developed a sensor to convert this language of volatiles into a language that one common man can understand. So here we'll give an example of the rice blight disease, which is a devastating disease in case of India and as well as the Asian countries. And there is very less or nil genetic resources available which provide the resistance. So what we have done, we have taken, this is the Swarna variety of rice infected with the Xanthomonas oryzae, which is the blight pathogens. And you can see into the lower figure that there's a visible symptom appears into the day 5. And what is our target? To detect the disease at day 1, day 2, or day 3, when a pathogen is there, but absolutely there is no symptom. Because once symptom appears, farmers are the best sensors to detect what kind of disease that are. So what we have done, we have specially designed a volatile collections chamber to directly trap the volatile that is produced as a result of plant microbe interactions. And we have optimized the parameters due to lack of time. I'm not going into the detail of that. And see, this is how these things are. Real time, these volatiles are collected from the plant. We are not disturbing the plant. Plants are infected. These are the solid phase microextraction cartridge, and directly it will collect the volatiles that is emitted during the plant micro interactions. And now we'll inject these volatiles into the gas chromatography mass spectrometry, and then we have seen what is the profile of a healthy leaf as well as the infected leaf. And you can see that red marked boxes, these are the volatile which are produced upon the infection of the plants to the blight bacterium. So we identified what are the nature of these signatures, and then we have gone for the multivariate statistical analysis, and we have identified 5 biomarkers which are produced as an early signature when there is a bacterial pathogen enters inside the plant, but absolutely there is no symptom. So this is the kinetic pattern of change of volatiles with the progression of the disease. And finally, we have identified that this LOX pathway, then there is a salicylic acid pathway, and carotenoid cleavage pathways are responsible for the biosynthesis of those volatiles. Again, I'm not going into the details of this. Then we have undergone the analyzing the gene expression level to support that whether this change into the volatile level is biosynthetically triggered. And what we have found that both the LOX gene and then the SMT1 and the CCT gene, which underlying the biosynthesis of all the 4 biomarkers, are upregulated just after the infection. Then, so we have identified 4 of these biomarkers as a target for the early detection of the disease. But after that, again, we started this journey to detect a biomarker for the early prediction of disease. Till now, the results are fantastic, but we are again coming into the 0 because this can be possible only with a very sophisticated instrument which costs in crores, and it requires a highly expensive and the long-term expertise. So what another option that is for the students, that the student can take in GC and go to the field. Of course, that is also not possible. So till now, entire outcome is 0. So that means we have an excellent biomarker, but to detect those biomarkers, you need a GC as well as you need a long expertise. And there the challenge started from 2017. Then we have developed transducers. So these transducers are the metal oxide based transducer which sense a particular volatile and resulting into the electrical signal change. So this is how this system works. So after this infection of the rice plant with the bacterial pathogen, it emits certain signature volatiles which react with this transducer and generates an electrical signal chain. And those electrical signal chains are monitored by very sophisticated circuits, and then this data is transferred through a Wi-Fi into the analyzing system. So this is the outcome of this is the coated fabricated device that is made up of metal oxides as well as the nanomaterial. And there are some cylindrical, carbon nanotubes are there to filter the volatiles as well. And this is, I will show another video how this system works. If you grow a plant inside a chamber, just you have to keep it for 2 minutes. Of course, these are the infected plant. And then you take the volatiles, put it into SPM vial, and go back to the field. Now, we'll inject into a device which has this coated sensor inside, and you can see there's change into the VI characteristic. So, based on the reaction of these volatiles that will react with this transducer, there will be an electrical signal change, and the proportion of the electrical signal change is proportionate to the change into the volatile concentration. Now you can see this volatile reacts and VI characteristics start to change. Again, the cost to measure this VI changes, you need a device that is 15 lakh rupees. So again, outcome is 0. So let's what we can do next. So fabricated a device which is very low cost, that is with integrated circuits and AI module. Then when you will go into the field and test the plants are of different size, timing different, cultivars are different, ages are different. So normal linear algorithm cannot handle this problem. So that's why we introduced the backpropagation artificial neural network to handle these decisions. And finally, what we found that till now, based on the 19,000 test data, the sensor can predict the disease with 85% accuracy. So this is how this prototype works. So just it's very easy to use, like different varieties are integrated based on the different agroclimatic zones. This is showing the plants are healthy, so absolutely there is no symptoms of the blight. I will take a second plant which absolutely phenotypically looks same. There is no symptoms of the blight, and now see how the sensor is predicting. The result goes down. So this is an N-type semiconductor, and it predicts that this is an disease. So now we are in a phase of automating this device into the field. So, and these all are patented technology. This is another sensor device patented which can detect the quality of the coconut without breaking the coconut. Again, I will, because of lack of the time, I'm not going into the details. And this is one of the topmost startup discovery that is recognized by the Inventive in 2024. And we have demonstrated into the University IIT Hyderabad. Then this is one sensor that can detect the scab disease in apple. Again, the condition is absolutely there is no symptoms into the leaves, and that is very instrumental for the apple producing state like Jammu and Kashmir and the Himachal Pradesh. These plants are healthy, so there's a green light, so farmers can easily look at into these systems. So see, the leaves are absolutely normal. So externally, you can't see that there's a disease symptom, but whenever you see take the device, it's called the scab stage 2. So now this sensor has been installed into the ICR CITH, that is the Apple Producing Institute at the Srinagar, Jammu and Kashmir. And this is validated with more than 80,000 samples. This is again patented technology. We have also developed sensor to detect the dry rot and soft rot disease of the potato. And at the end, what is our aim? Like we have in protections, we have in doctors, we have in engineers. So our group is aims to develop a protection layer made up of sensors. So plant can sense either a biotic or a biotic stress through an additional layer of the sensors that our group is dedicated. Now, I am very thankful to the generous and extremely well funding from all the funding agencies so far available in India, especially for the ICAR, NSF, for routinely supporting me in this research, Coconut Development Board, DST, DBT, STAR, SCAN, and lots more. These are my all external collaborators outside the India, and inside India there are too many. Two of them are sitting just in front of me. And these are my students, so they should be thankful to me for presenting their work on behalf of them. This is our institute, so this is very close to your Amity. It's just 3.5 hours. If anybody wants to do it or get an exposure of sensor research, you are welcome at IIT Roorkee. And with this note, I will end my presentations. If you have any questions, I will be happy to answer now. Or since this is a lack of time, I am available, so you can always ask me after this talk as well. Maybe during tea. They are in hurry. Yeah, okay, thank you. Thank you.
Subject. Thank you. Sir, आप लगा दीजिए मैं साथ में बोल देती हूँ. Moving forward, I invite our next speaker, Professor Dr. Sriram Yadav, sir, Department of Biosciences and Bioengineering, Indian Institute of Technology, Roorkee, India. Sir has completed his postdoctoral fellowship with Professor Hela Rota at Institute of Biotechnology, University of Helsinki, Finland. Sir has published over 50 research papers in various journals of repute and has 5 patents to his credit. He has been invited as speakers to various national and international seminars and workshops. He is also the editorial board member for Plant Growth Regulation, Springer, guest editor for Plant and Cell Physiology, Oxford University Press, and guest editor for Springer. He is also a fellow of National Academy of Sciences, Prayagraj. Thank you, sir, for joining us on AMIFOST 2025. I request you to kindly share your experiences and your knowledge with our students.
Good afternoon. First of all, I would like to thank the organizer, Dr. Renu, Dr. Monica, Dr. Durgesh, for giving this wonderful opportunity to share some of our work which we are doing at IIT Roorkee. So as you can see, I come from the oldest technical institute of this country, or Asia, IIT Roorkee. We have recently completed 175 years of our existence. Is there some pointer? Sorry. Is it something pointing? No? Is there a pointer? No, it's okay. No problem. No problem. So today I'm going to just present some of our work where we are trying to understand, can we use genome engineering platform to alter plant architecture to improve yield and nutrition. Thanks to Professor Mukesh. So I don't have to give much introduction about the importance of these things, importance of these works, particularly for food security and in this growing population. So everyone is contributing as per their expertise. Being a developmental biologist, being a genetist, we are also trying to use our expertise to understand. Okay, great. So plant architecture, architecture of any organism is very, very important. And architecture is a key determinant of yield and adaptability for any plant. So the way you shape your structure, it is very crucial for you to be your plant. If you look here, this is the modern dicot plant Arabidopsis, and then you have rice, which is basically economically important crop. You can see they have a different architecture, shoot architecture, everything is different. Even if you look the inflorescence, which is actually producing grains, you have a huge difference in the architecture. So our question is that, can we modify the architecture and does it have any significance to improve the yield and nutrition? And there are some reports, if you look, this was a paper in Nature Genetics, where they identified a mutant, and in that mutant, just there is a change of a little angle of the milk formation. And that change here, if you look here, it generates a plant which is more deeper penetrating roots. And it gives up to 70% of the yield benefit under drought conditions. So which means that the architecture is a very, very important. If a plant has a better architecture, it can sustain in a harsh condition better way. And that is definitely going to affect the productivity. What we are doing at IIT, we are doing a lot of fundamental research. And we are trying to understand the molecular mechanism which determines the architecture of the plant. And then we take this fundamental research and try to generate a proof of concept towards translational work. Can this have some benefit in the plant towards the yield and nutrition? And here I'm going to talk primarily on the root architecture. Root is one of the most important component of plant, I would have to tell you. And we are using rice, which is economically important plant, as a trait where we are studying. So if you look at Arabidopsis, as I said, this is the dicot. In dicot, you have a taproot system where you have a primary root, and then there are a lot of lateral roots coming. But if you go to the monopod, particularly crop, you don't have primary root and lateral root. But apart from that, there are a very specialized root, which we call adventitious root or crown root, which originates from the shoot. And what is the fundamental difference? Primary root originates from the embryo. Lateral root originates from the root. So they are root-borne root or embryo-borne root. But adventitious root is the only root which originates from the shoot, which means that there is a shoot tissue after receiving a proper signal, it switches its lineage and it starts a root-specific developmental program. So if you make this section, cross section at the stem base, you can see this is how it goes. So these are the root primordia. You can see 5 root primordia at different developmental stages. And they are initiated by a ring of this that ring we call competency. And if you look, also the competency is everywhere, but roots are initiated only in a localized domain, which means there has to be a domain-specific signaling that need to be activated for initiating the root formation. And we are trying to understand what are the molecular mechanisms, what are the key genetic regulators of these processes, how these processes are being initiated at the very first stage. For that, we have used a highly sophisticated technique which is called laser capture microdetector. In this technique, you can very specifically see that we have collected these tissues, the primordia at early stage, initiation stage, as well as when primordia is outgoing, and we try to do RNA sequencing. And what we have done, we have identified all the genes which are very specifically and sharply getting activated when shoot tissues are changing their fate from shoot to primordia. That is the crucial moment. That is the triggering moment. Once the decision is taken place, then nothing happens. It cannot revert back to shoot again. It will have to initiate root and it will come out. I will not go into all these details because these are genomics and you go. understand this very well. I will just show some of the examples. If you look, these are some transcription factors, and all these transcription factors, if you look, their expression in the control tissues or in the competent tissues, their expressions are very, very low. And if you look all these genes, their expression suddenly shoots when primordia is initiated. And then later on, they have a different expression. And we perform RNA in situ hybridization to validate this expression because RNA sequencing data need to be validated. You can see here in the cross section of the stem, and you can see all the root primordia. When the first two cell division is started, you can see these genes have started getting activated. It suggests that these genes are getting activated exactly at the onset of primordia initiating. Okay, so there could be a potential regulator. Then we ask the question, okay, do they have some functional significance or they are just present here? If they have some functional significance, they should show something of that. So we looked the literature, there was some gene therapy called VOX. In VOX, if you look, there are two VOX genes in Arabidopsis, and when you double mutate them, if you do the single mutation, there is absolutely no effect. When you make a double mutate, you can see that root organogenesis is affected. We looked in rice, and what we found that in rice there were 3 genes from this family, and there was already literature available that one of these VOX genes, when you downregulate these, you can see there is no root formation or alveolar root formation, and when you overexpress this gene, you can see that roots are coming from the shoot, even from the leaves, roots are coming. So we took one of these genes, and why we took, if you look the expression in our laser capture detection, this gene was almost 0 in the competent tissues. Then expression goes up and then again comes down to 0. This is very, very interesting. The pattern says that it might be triggering a process and then it is no longer required. So this is commented like a director of your institute, comes in a conference, gives 5 minutes speech, and then goes. Process is started. Now we don't see director to be present throughout the seminar. So this is the master regulator. This was, and we validated. How we validated? When we down regulated this gene, you can see that root initiation was. At 4 days, you don't see any initiative. In fact, when we use CRISPR and generated knockout, you can see at the very next stage that in case of mutant, number of adventitious roots were decreased. Very interestingly, if this is a triggering factor, what you are expecting, it should initiate a process. And if it is no longer required, you just give a pulse induction. You overexpress this gene only for 24 hours, only for 12 hours. Then don't remove the expression. Can it initiate the process and process happens? Then answer is yes. Look here, if I don't stop, it will continuously start doing because the master regulator is already activated. Now it will not require anything else. Process has started. In fact, its presence are going down at the later stages required. So that is where this becomes a very master regulator. If this is the case, we look how conserved this gene has a function. So we take this rice gene, which is a monopod, and put in Arabidopsis. Arabidopsis is a dicot. Dicot has an absolutely intact root system. Monopod has a fibrous root system. Very different kind of root system. And we took one single gene of rice and put back in Arabidopsis. Can you see here, in fact, Arabidopsis mutants as well. You can see that Arabidopsis root system looks like fibrous root system. So evolutionary, if you look, I can say it is a very big conclusion, needs to be validated every day. But if you look here, this is the back root system, this is the fibrous root system. Now my tank root system has started looking like a fibrous root system just by overexpressing one single gene, which means that this gene might be a gene which was evolved when there was a diversification of tank root system, fibrous root system from the tank root system. And it was evolved at the time of fibrous root system formation and continued its process and probably has a different function in type of system. Similarly, we look another transcription class of transcription factor. This is plethora. In adoption, this is known very less than 5 transcription factor. If you look at triple mutate, you can see there is absolutely no lateral root formation. Lateral roots are initiated, but they never come out. We selected some of the plethora from rice because we could find a large number of plethora which had a very strong expression. Look here, one of these plethora Okay. When we knocked out, you could see that root initiation was very interesting. So when you generated this for knockout, all the three, everything, you can say that would positively affected. On the other hand, when you overexpress this gene, when you increase the expression level, you can see both adventitious root and lateral root number will significantly increase. So the cross section, you can find a lot of difference. When you continuously increase overexpress, continue express, you can see that root has started coming from the tip of the leaf, which means that this gene has the tendency to activate root formation from shoot tissue anywhere. Yeah, there is a tag which I'm not going to tell you at this moment, but you believe that it has a tendency, it can activate if I do little bit. That little bit I will tell sometime later. Okay. Similarly, there are some other plethora genes which you can see here. So what I'm trying to say here that we have a method through which you can identify the key master regulator of root architecture. Different. Another interesting thing here, we do the same thing. If you look at the mutant of the rhodopsin, there are lateral root primordia initiated, but these lateral root primordia are not coming out because of the absence of plethora. We take one rice plethora and put back here very specifically only in the arrested primordia to see all the lateral root primordia had started coming out. So irrespective of the fact whether it is from dicot, it is from monocot, how diverse is the species, but their molecular function in promoting root outgrowth is control across the planet. Okay, Auxin, you all know Auxin is a master regulator of root formation. Another project we have been doing with Mukesh and since we started the lab and we identified all the genes, proteins, metabolomics, everything which is regulated by Auxin signaling. We selected a couple of transcription factors from this study and we studied and we see the new things which is absolutely new data for to present here. You can see here we found one of these transcription factors which has a very interesting expression pattern in the primordia. And when we are giving Auxin signaling, the primordia, you will get that the expression is getting restricted at the boundary of primordia. And when we generated knockout of this gene, this was the interesting component. When we mutate it at the early stage, what is happening? The process of root initiation is delayed. When we did a very careful analysis, we could found that only first two roots or first two adventitious roots are delayed. All other roots are absolutely normal. This is the plot. If you can see root wise, you can see. So if you delay in initiation of first two roots, then eventually all other roots are at the normal. When you make a cross section, we ask this question whether it is because of the initiation delay or all those roots. Initiation happens absolutely on right side, wild side mutate. But if you use the outgrowth, the first two primordia in wild type, it is going faster than the root. It suggests that we have identified a very interesting gene which has a very specific function. But when we continue to grow this plant later on, we observe something more interesting. And if you look here, this mutant it has a longer system, deeper penetrating to the system. Then we ask the question that it has some genetics to the benefit of the yield or nutrition. And when you look this plant grows robustly very healthy and it is very similar to the wild type morphologically. So there is no bad effect of this. But when we counted the seed number, when we look the yield parameters, we can see that the length of seeds was slightly higher. The weight was slightly higher, which we will provide data. And look here. So here, if you take the 50 seeds of white type and you can see both length as well as weight is significantly, marginally but significantly better than the imputed than the. And beauty here is that this is loss of function. In loss of function, you can use CRISPR and you can generate a marker-free line, transition-free line. The transition-free line is more important. Then we collaborated with Professor Khan, and what we wanted to see that is there any change in the nutritional profile of this grade of the mutate. And what we can observe that, in fact, the right grade which lacks most of the amino acids. And we could see that in the nutrient background, some of the important key amino acids are getting increased. So this suggests that It is a kind of the general proof of concept that the ultra-business architecture, even the nutritional parameters, not only three, is as well as nutrition, both can be beneficial. Apart from that, these are some other mutants where we are now expanding our study and we are looking more branching events like anical, inflorescence, wherever we see the field parameter. You can see there is another mutant where we have generated. We can see here there is a more branching of the shoot. So in rice, typically one plant is one branch. One branch means one inflorescence means only that nothing. You can see here in this mutant background, one plant has 3 or 4 branches, and all 4 branches are making panicles. All 4 branches. So one plant is giving more. Of course, there is another catch which we still have to do. But this is a proof of concept that we know some crucial genes. If we manipulate that, we can actually change the architecture of the plant, even shoot architecture as well as the architecture. So this is what I wanted to say. This is my team who has done all this work. So as they said, we are here just to present their work, nothing else. They are doing most of this work, almost all this work. These are my collaborators, past lab members, current lab members. These are the funding bodies. And of course, in any research, you require 3 things: your idea, manpower to working, and funding. And all 3 are equally important. And I have been lucky that I found all 3 at right time all the time. Thank you very much.
Thanks, sir. Any questions? Any participant, student have any question to the sir? Thank you, sir. Okay. We move towards our next speaker she has joined us online from Portugal, Professor Dr. Ana Santos Silva, Faculty of Pharmacy, University of Coimbra, Portugal. Professor Silva holds a degree in pharmaceutical sciences from the University of Coimbra and earned her European PhD in pharmacy with honors from the University of Santiago de Compostela, Spain. Dr. Silva has a remarkable track record with more than 250 scientific publications. Her research focuses on the development of innovative packaging technologies such as active and edible packaging and the study of packaging migration into food. He has a strong interest in analytical methodologies, particularly mass spectrometry for detecting bioactive compounds, food contaminants, and packaging migrants. In recent years, she has also explored natural extracts from plant and food byproducts for their integration into active packaging systems. Throughout her distinguished career, Dr. Silva has received over 20 research awards recognizing her outstanding contribution to science. In 2024, she was honored with the prestigious Food Packaging Award in honor of Dr. Reister Rees Davis and Darren Brody by the Institute of Food Technology, one of the most highly regarded awards in the field of food packaging globally. Additionally, Dr. Silva has consistently recognized among world's top 2% of most cited scientists in the Stanford University ranking since 2021. Thank you, ma'am, for taking time out from your busy schedule and joining us straight from Portugal.
Thank you so much for the presentation. Thank you also to the organizing committee for to attend this conference and to be able to speak a little bit about the work that we develop in our faculty. So it is a pleasure to be here. Unfortunately, it is not possible in person, but at least we are able to be in contact online. So today I'm here to talk about the future of food, also about sustainability, safety, and technological innovation. But first of all, I would like to present you my home city, Coimbra, which is in the center of Portugal. And also I would like to present the University of Coimbra, which is the oldest in Portugal, one of the oldest in Europe, and it is UNESCO World Heritage. Also, I would like to show you some photos of our faculty, the Faculty of Pharmacy. This is our building, and also there is a very strong academic life in this faculty. So in general, the students, they really enjoy to live the experience of studying in this faculty. So now returning to the subject of this presentation, of course that when we are today talking about the food system, we cannot just talk about food chain from the producer to the consumer. We also have to look to all the factors that influence this chain and are influenced by it. Also, to talk about all the entities, institutions, and people that is involved, and of course the connection regarding and among these elements very important. So we should talk about food systems instead of food chain, and food systems are very much more complex. So we know how important the food system is, and the influence that has in order to avoid the dramatic consequences of climatic changes. So these climatic changes are threatening the production, processing, and distribution of food. And we know that in the future years, we need an increased amount of food to feed all the world. So in 2050, we predict that there is a demand of about 50% higher amount of food. So we need to revise the consumption patterns. Also, we need to reduce the food waste and the food byproducts in order to feed all the world population. So there are many challenges in the food area, and here in this slide we also only show the main ones. Of course, we cannot forget about food safety, also the ingredients of food, because food industry wants to replace some ingredients that are considered not so healthy by healthier ingredients. Also food additives, and there is a major concern regarding food additives. We also intend to substitute some synthetic food additives that are related to adverse health effects by natural molecules. Of course, that the quality control is very important, and there are many parameters that we really have to monitor. Sensorial analysis is of utmost importance, because it's related to the organoleptic properties of the food. So these really need to be of high quality in order the product can be accepted by the consumer. But also we need to check for sustainability, also traceability, big data, and of course it is nowadays very important also the consumer awareness regarding food safety issues, but also regarding the relation of food with health and well-being. So on the top of the trends in food area, we have sustainability and economy, circular economy, but also there is a trend of trying to change the list of ingredients of food and have clean label food food, which the listed ingredients, it's very easy to understand, and generally it corresponds to foods that are less processed. Also, there is a trend of using 3D printing to achieve unique food regarding their appearance, but also regarding their composition. Also in Europe, we are not very used to incorporate insects in our food, but now there are already some companies that are producing insects specifically for food purposes. Also, lab-grown meat, it is a tendency, although some limitations like the high price. Functional foods are also very trendy, and of course also the change regarding the food packaging. So we tend to substitute the conventional polymers that are not biodegradable by those that can be obtained by renewable sources and or are biodegradable. So we tend to incorporate bio packaging and also try to give it a further advantage when they incorporate bioactive compounds that can migrate to food. So we are talking about active packaging. So regarding the food sustainability, of course, that's the word sustainability is applied to many sectors, also to the food sector, and it tends that we meet the nutritional needs, but without compromising the future generations. So we want to reduce the environmental impact of the production and of foods. So generally we tend to talk about foods that are produced at local level, are seasonal and produced with methods that respect environment, animals and consumers. So in this regard, in our lab, we carry out many analyses in order to develop methods to evaluate mycotoxins, for instance, in food items. Of course, that when we talk about mycotoxins, there are so many factors that affect the proliferation of these toxins in food that some are related with the field, some are related with the transport from the field to the supermarket. Others are related with the type of matrix, of food matrix. For instance, the dry foods, depending if they are peeled or if they are dried, or the drying conditions can very much influence the contamination with mycotoxins. Also, the sampling is very important, as well as any processing method that can be applied. So in this regard, in our laboratory, we decided to optimize a method to extract several mycotoxins simultaneously. So one of the tests was regarding to optimize the solvent that we were going to select for the extraction. So these are the results comparing aflatoxins with other mycotoxins like ochratoxin A, zearalenone, toxin T2 and HT2, and also 2 fumonizins. And we could see the differences when we acidified the water with formic acid. So we decided to test different percentages of formic acid, and we could check that for aflatoxins, the increase of formic acid was not a good choice, but on the other side, the incorporation of formic acid in the ultra pure water used for the extraction process, in the case of fumonizins, could enhance considerably the extraction of these mycotoxins. So depending on the properties of the analytes, we should select carefully the solvent, and in this case that we want to analyze all these mycotoxins at the same time, we should we decided to select an intermediate solvent, not the solvent that achieved the best results for aflatoxins or the one that achieved the best results for fumonisin. We decided to select water with a very low level of formic acid, with 0.1% of formic acid. Okay, also we use a Quetcher's method, a method that is considered cheap, easy, effective, rugged, and safe. Of course, this method, it is generally applied for pesticides residues, but we decide to apply also for mycotoxins, okay? And we did different tests in order to optimize the best composition or the best mixture of salts to be used, and also the best absorbent to be used, okay? So here we have some of the results. We tested many conditions, and of course we did not achieve the same results for all mycotoxins, because these mycotoxins, they have very different physicochemical properties. So just to check, two of the best absorbents were EMR lipid and ZSEP. But of course, although for some of the molecules we could achieve similar results, in the case, for instance, of fumonisins, they were not detected when we were using ZSEP. So we decided to validate two methods. So one method using in the extraction the absorbent EMR lipid, and another method with ZSEP absorbent. These are the results of the validation parameters. So you could see the differences regarding the detection and the quantification limits using both methods, okay? Of course, that if you really need to detect these two fumonisins, you should, of course, use the method that has or that uses EMR, and the other one does not work for these mycotoxins. Also, we evaluated the matrix effect. As you can see, most of the results were between the limits of 80 till 120% of matrix effect, with an exception. Ochratoxin A presented a very high matrix effect when using EMR, or a very low matrix effect, a suppression effect when using the ZSEP. Then we decided to apply to some samples. We decided to select a dry food that generally it is recognized for being contaminated with mycotoxins, which is pistachio. Okay, so in one of the samples we detected aflatoxin B1 with 0.2 micrograms per kilo. Also, we detected in another sample, but in this case of a byproduct of pistachio, we detected HT2, okay, the level of about 50 micrograms per kilo. So we decided in this work to evaluate not just the samples, but also the byproducts of these samples, because we wanted to know if we could use this byproduct to valorize it, and also to check if it was we were able to reuse for food purposes. Another analysis that we carry out in our lab is regarding the plant toxins like terpene alkaloids. Okay, we know that this subject is really relevant at the European level. Okay, there are many types of plant toxins. Some they appear in edible groups, other in toxic plants. And our work was, in this case, regarding terpene alkaloids, especially two molecules, atropine and scopolamine, because they have anticholinergic effects. So they can, people that eat and consume foods with high levels of atropine and scopolamine, they can have blurred vision, dry mouth, tachycardia, but this at very high doses can also lead to convulsions or even death. So this is one of the plants, the Thura stramonium, that can contaminate, for instance, cereals and originate problems. In this work, we decided to evaluate plants, dry plants that are used for infusions, okay? So we also used a Quetscher's method that we have changed a little bit in order to optimize specifically for these compounds. We carried out the determination using an UHPLC with a time of flight mass spectrometer. And besides atropine and scopolamine, that are just the two molecules that have the regulatory limits at European Union level, we decided also to incorporate anisodamine and ometropine in our method. Okay, so here we evaluated many samples. And we found atropine in a Melissa officinalis plant and ometropine in Urtiga dioica. These are just some examples of samples that we determined. Also, I would like to talk you about the active packaging. I work a lot in this field. And when we are talking about this type of packaging, we want that the packaging the packaged food. So we can have a group of active packaging that releases compounds to the packaged food and the intent, or we can have absorbance, a group of active packaging that is going to absorb a compound from the packaged food or from the atmosphere inside the packaging. The aim is always the same, to increase even more the shelf life of the product. So in this regard, I work a lot with extracts that I obtained from plants or from food byproducts like fruits or vegetables. So I incorporate in a polymer, and then I can develop active releasing packaging that will release these compounds to the packaged food during storage. So when doing this development, we have to take into account the activity of the extract that we are using, but also the polymer that we select will have a very important role in the effectiveness of the final packaging. Also, of course, the percentage of incorporation is important, as well as the properties of the food, if it has very high fat content, if it is liquid or solid. But also the temperature of storage is one of the factors that influences the final effectiveness of this type of packaging. So this is an example of some citrus byproducts that we decided to test. We have contacted a fruits company fruits juice company. So these are industrial byproducts. We test them regarding the antioxidant properties. In this case, we were comparing orange, lemon, and lime pomace. And lemon and lime, they were the two byproducts that presented the highest antioxidant properties. Okay, so we also studied the profile on phenolic compounds. We started with 27 phenolic compounds. Nowadays, the method has more than 50. And we also use our equipment, the UHPLC time-of-flight mass spectrometer, to carry out this evaluation. In these byproducts, eriocitrine and sperridine were the two phenolics that were found in highest amounts. Okay. And we know that there are many factors that affect the antioxidant properties of these extracts. Of course, the byproduct that we select, but within the byproduct, also the cultivar or the variety, also the edapho-climatic conditions are very important, as well as the part of the plant or even the maturation state when we are talking about fruits. Okay. It's also very important to check the storage conditions as well as the extraction method and the method to determine the antioxidant properties. We are carrying out a project, an European project, where each of the partners has selected different byproducts. These are just some examples of byproducts that were selected for this project. But we decided not just to evaluate the antioxidant properties, but also potential contaminants like mycotoxins and pesticides residues. So for pesticides residues, we have a method that allows to determine more than 150 analytes. We used a Quetscher's method, okay, and we have a low limit of quantification of about 5 micrograms per kilo. So these are the results, some of the pesticides residues that we found in our byproducts. As you can see, cherry seeds were the samples that present more pesticides. Okay, date seeds they present were the only samples where we did not detect any pesticide. So we were worried because these are the results in fresh byproduct, and when we are preparing the extracts, we are concentrating the extracts on their content on bioactives, but we could also be considering some of the contaminants. So we nowadays have also a method to determine pesticides residues in extracts. So we miniaturize the method. So right now we don't need 10 grams of sample to do the analysis. We just need 100 milligrams of sample to check if the extract is or not contaminated with these contaminants. Also, we evaluated mycotoxins. In this case, we were able to detect all the mycotoxins that have legal limits in the European Union, but we just detected ochratoxin A in a very low level, lower than the limit of quantification of 1.5 micrograms per kilo in one of the samples of date seeds. So we have to balance what is the bright side of using plant-based extracts, because we really can extract from plants and fruits and vegetable byproducts some antioxidant compounds like carotenoids or phenolics, Also, we can extract some natural antimicrobials, but we cannot forget that we should also evaluate potential contaminants. So we have many advantages when we're using active packaging. We can increase even more the food shelf life, substitute some synthetic additives by natural additives, and also to reduce the amount of additives that we add directly to a food item. Of course, when we incorporate these extracts in biopolymers, we can also reduce the environmental impact of packaging as well as the costs of food waste management. But we have limitations. We know that some natural extracts can be contaminated with some contaminants, so we shall evaluate this level of contamination. Also, when we are talking about antioxidants, we might find some pro-oxidant effects, so we should not use very high amounts of antioxidants. And in Europe, we also have regulatory limitations, so we need the approval of our authority, EFSA, in order to have the extracts that we want to incorporate in the polymers to be authorized. So in the future, we really need to standardize the quality of the natural extracts in order to guarantee the quality and the effectiveness of these products. Just to finalize, I want to show you some of our papers, recent papers on active food packaging, and also I would like to thank all the colleagues that collaborate with me in these studies. Also to thank the project that is founded these studies, and just to thank you all your attention, and to say that I'm available to any questions that you might have. Thank you so much.
Thank you, ma'am, for such an overwhelming session. I request our session chair, Dr. Neerja Zola, ma'am, to kindly sum up the session. So thank you so much. It was a real pleasure to chair this session. We had some very, very interesting 3 presentations in this session. The first presentation by Professor Sarkar talks about artificial intelligence. We all know that that's going to be the next big boom in the world, and how he's used sensors for detection of infection in plants by different pathogens, even before the plants show any kinds of symptoms. Then we had a very nice and presentation by Dr. Das, where he has actually shown how just manipulating genes can actually change the architecture of shoots and roots. So that was really interesting. And the third, all the way from Portugal, about active packaging and how extracts can be used to increase the shelf life of food products and possibly reduce the use of additives in food products. But of course, they are subject to regulatory approval. So thank you very much. It was a very interesting session, and I was honored to chair it. And I would like to thank the organizers again for this kind invitation. Thank you so much. Thank you, ma'am. I may request our HOS, Monica ma'am and Dani ma'am, to come on stage and kindly felicitate Dr. Neerja Zola ma'am, Head Science and Regulatory Affairs, Yakul Danone, India Private Limited, Delhi. I request students to give a big round of applause to our session chair. I request HOS sir and Neerja ma'am to remain on the stage. and kindly felicitate Debarata Sir, Bioscience and Biology Department, IIT Roorkee, with a memento. I request Junior Sir to be kindly be on stage, sir. Please join us on stage. Durgesh Sir, please kindly be. I request our dignitaries to be on stage and kindly felicitate Dr. Sriram Yadav Sir, Department of Biosciences and Bioengineering, Indian Institute of Food Technology, Roorkee, India, with a memento. I extend my thankfulness to Professor Dr. Ana Silva, Faculty of Pharmacy, University of Coimbra, Portugal, for joining us for the session for the 8th AMIFOST 2025. Thank you. So, sir, I request HOS sir to be on stage. I request HOS sir to kindly felicitate Dr. Neerja Hajela, Head Science and Regulatory Affairs, Yakul Danam, India, for such a heartfelt sponsorship. I request ma'am's team to be on the stage. Means we need are going to for the in the same venue F2 auditorium, and we will be joining for the networking. Okay, session 3, the technical session 3, harnessing nanobiology for agri-food technology will take place online. I request the participants and the students to kindly join through the link as provided in the program schedule at 6 p.m.
Begin within 3 minutes. We will start the session. Professor Sivendra Sahi will also join the session within I think 2 or 3 minutes. He will join. You are welcome, Professor Lugia, to our conference. Thank you so much for joining us. Nice to see you, all of you.
Good evening, Durgesh sir. I have joined. So you are muted.
Okay. Yeah, we will start. We are just waiting, Professor Sahi. I think he's also joining.
Okay, sir.
Hello.
Yes, sir.
Please, now I think our session chair has also joined the session. So please start the session.
Okay.
Hmm.
Good evening, everyone. So we have come to the last session of the 8th AMIFOST 2025 International Conference on the future of food science and technology, innovation, sustainability, and health. And we are starting with the theme harnessing nanobiology for agri-food technology. I request Dr. Durgesh Tripathi sir to kindly welcome our session chair, Professor Shivrendra Sahi, to our participants.
Okay. Thank you so much, Dr. Sharma. So, distinguished guests, esteemed speakers, and scholars, a very good evening to all distinguished faculty members and students. With the kind blessings of our Honorable Founder President, Sir, Chancellor, Sir, and valuable guidance of our Honorable Vice Chancellor, Madam, it is our privilege to welcome you to this highly anticipated session on day first of the conference organized by Amity Institute of Food Technology. We are deeply welcome to Professor V.K. Modi, sir, professor and head of the Amity Institute of Food Technology, and also the chair of this international conference. I'm also welcome Professor Monica Thakur, Madam, the convener of this international conference, and Professor Renu Madam, organizing secretary of international conference, and also some other faculty members who are the board member of this international conference. I extend my heartfelt gratitude to our distinguished guests, esteemed speakers, and enthusiastic participants who have gathered here this evening. Your presence enriching this occasion, making a hub for the groundbreaking discussion, innovation, ideas, and meaningful collaborations. We are privileged to have Professor Sivinder Sahi from St. Joseph University, Philadelphia, as our session chair. His vast expertise and leadership will undoubtedly set the tone for an engaging and thought-provoking discourse. I also would like to welcome our other invited speaker, a keynote speaker of this session, Professor Jason White, from the Connecticut Agriculture Experiment Station, a global authority on nano-enabled agriculture, food safety, and environmental sustainability, delivering tonight a keynote address. I also like to welcome Professor Renato Grillo from São Paulo State University, Brazil, a renowned expert in plant protection, biotechnology, and sustainable agriculture practices and nanotechnology. Also like to welcome Professor Lugia Medelo from Federal University from Brazil, a leading researcher plant secondary metabolites, nitrogen nutrition and stress response, and also in nanotechnology. Each of these esteemed speakers bring a wealth of knowledge, innovation, and inspiration to this platform. Their pioneering research continues to redefine the frontiers of science and technology in agriculture and nanotechnology. Additionally, we conclude we will feature oral presentation by research scholars. After their presentation, showcasing their cutting edge research that is shaping the future of our nanotechnology field. Before we begin, I once again extend my deepest appreciation to all our speakers, guests, and participants for making this event possible. Your presence and engagements make this gathering an enriching experience. Let us immerse ourselves in this session with curiosity, enthusiasm, and spirit of learning. I look forward to an evening filled with the insightful discussion, transformative ideas, and meaningful collaboration. So, wishing you all the fruitful and inspiring session, and wishing you all welcome in this very meaningful nanotechnology in agri and food technology session. You all are welcome. Now, I'd like to hand over this session to Dr. Sharma for further proceedings.
Thank you, sir. It is with great pleasure that I extend a warm welcome to all of you and to this special gathering. Today, we have the honor of welcoming a distinguished scientist, an accomplished academician, and a pioneer in the field of nanobiotechnology, Professor Shivendra Sahi. Currently a faculty member at St. Joseph University, USA, has made remarkable contribution to the field of biological fabrication of nanomaterials and their potential applications. His research in nanotechnology, particularly in evaluating the environmental impact of titanium, gold, and silver nanomaterials, had been widely recognized. With over 155 publications, 8,623 citations, and an impressive H index of 50, his work has had a profound impact on the global scientific community. His accolades includes the Outstanding Researcher Award, the Career Professional Award, and editorial position in esteemed journals such as the Journal of Plant Physiology and Biochemistry and the International Journal of Environmental Sciences and Technology. Furthermore, his contribution extend to patented innovation in the eco-friendly synthesis of gold nanoparticles. Today, we look forward to hearing from Professor Sahi, gaining insight into his groundbreaking research and exploring the exciting frontiers of nanotechnology and its application. His presence here is truly an inspiration for all of us, and I am sure we will benefit immensely from his expertise and wisdom. With that, let us all extend our warmest welcome to Professor Shivendra Sahi.
Professor Sai sir, Professor Shivendra Sai sir. Dr. Sharma, I think Professor Sahi is in the meeting. So now we can invite Professor Jason for the keynote speaker, and later he will start. Ji sir.
Hello, I'm here.
I think he is facing some technical issues. So now we should invite Professor Jason White for his keynote lecture. So please introduce first, and then you invite him.
So Sarah, I should start invite the other speaker.
Yeah, Professor Jason, you can now
Okay.
invite Professor Jason for his keynote speaker.
Okay. We have a pleasure of hosting a world-renowned scientist, an exceptional leader in sustainable nanotechnology and agricultural innovation, and an esteemed researcher, Dr. Jason White. Dr. White is the director of Connecticut Agricultural Experiment Station and has dedicated his career to advancing nano-enabled agriculture, ensuring food security, and mitigating environmental risk. His groundbreaking work in nanotechnology, phytoremediation, and food safety has significantly contributed to our understanding of the fate and impact of nanomaterials in agriculture. With an outstanding academic record, a PhD in environmental toxicology from Cornell University, and a career spanning over 2 decades, Dr. White has established himself as a leading expert in agricultural nanotechnology. His impressive research portfolio, supported by over $5.1 million in competitive funding, has resulted in more than 500 publications, over 33,000 citations, and a remarkable H index of 94. Beyond his scientific contribution, Dr. White holds prestigious positions as a visiting scientist at Howard University, an adjunct faculty member at the University of Massachusetts and the University of Texas El Paso, and the immediate past president of the International Phytotechnology Society. He is also an editor and advisor for multiple leading scientific journals, shaping the general discourse on environmental sustainability and nanotechnology. It is truly an honor to have Dr. White with us today. His insights and expertise will undoubtedly inspire us all and provide invaluable knowledge for advancing sustainable agriculture. With that, let us extend our warmest welcome to Dr. Jason White.
Hello, can you hear me?
Yes, sir, we can hear you.
Yes, we can hear you, Jason.
Okay, let me share my screen. Does the screen look okay?
Yes, sir.
Yes.
See if I can get it in presentation mode. Okay. Well, hopefully that looks okay. Thank you for the invitation to speak. I'm actually in California right now in the United States at the American Chemical Society meeting. So it's approximately 5:45 in the morning here. So it's a bit early.
Yes.
I'm thrilled to participate in this meeting. So what I thought I would do is kind of give a brief overview of some of the work we're doing at the Agricultural Experiment Station right now on nanotechnology in agriculture, and hopefully set up some discussion for the remainder of the session. So the Agricultural Experiment Station in Connecticut, we are actually a government agency. So you can think of us as basically a research laboratory. We have about 110 staff that work for me, working on different areas, but obviously nanotechnology and agriculture is a big part of that. So this kind of gives you an overview of why we think agriculture is an area that's in need of significant research. This is from a couple of papers that we published in Nature Nanotechnology over the last few years, where we kind of laid out what we see as the current issues in agriculture. Let me see if I can get my pointer here. Yes. So obviously we know we had the Green Revolution, 70 or 80 years ago that significantly improved agricultural production. But we also know that conventional agriculture is not sustainable and it is not sufficient for the needs that we have in the future. There's a couple of different ways to look at this that we laid out in this first paper here. But one of which is 70% of all fresh water use on the planet goes to agriculture every day. That is completely not sustainable. We also know that in this paper, we looked at actually 11 different crop species. And what we found was that over the last 4 to 6 decades, depending on which crop you picked, the year-over-year productivity for every single crop was decreasing. The orange bars are the older 1975 to 84. The blue bars are 2005 to 2014. So this is just four of the crops, but clearly you see our ability to produce food is decreasing over time. We also know we have significant problems still globally with hunger and hidden hunger or micronutrient deficiencies. We know that in spite of significant pesticide use, we're still losing 20 to 40% of our crops to disease and pest. And then this other issue, which is where we actually started to work about 10 years ago. The efficiency of pesticide, fertilizer, stimulant delivery is really, really low. 75 to 99% of what's being added to the field is what's being added by a grower to the field is not making it to where it's needed. And to be honest, this is actually what got us thinking about nanotechnology about a decade ago, because we in agriculture, we were watching what was happening with medicine and with nanotechnology in medicine. And with varying degrees of success, what was happening there is nanotechnology was being used to deliver cargo very precisely in a complex system. That complex system was the human body. But those of us looking in agriculture, we looked at that and kind of saw a similar problem. And we needed to increase the precision of delivery of our cargo, which instead of being medicine was fertilizers and pesticides, in a very complex system, which is, an agricultural field. So it's not a perfect metaphor, but it kind of got us thinking about what nanotechnology could potentially do. But this is obviously the real motivation for why we're having this session and why there's so many people interested in this area. And we've known this for a decade. We've known that the conventional agriculture is going to need to change. We're going to have to increase food production by 60 to 100% by 2050. And to be honest, it's probably by 2040 at this point that, we're moving in the wrong direction. And remember the data we were just looking at in the previous slide. So we've been moving in the wrong direction in terms of productivity for literally decades. And so we're going to need to turn that around dramatically. And it's not going to be easy. We know that climate is changing more quickly than we thought it was going to, and this is creating greater stresses. And then there are human issues. Humans have a tremendous ability to get in our own way, and that's certainly happening here. So many of us who work in this space, and I think many of us who are on this in this meeting, we know we need to completely have a transformation in terms of how we grow, distribute, and store food on the planet. And that nanotechnology can actually play a significant role here. So these are just some of the areas that we've been working on. I'll show you a few examples of each over the next 20 minutes or so. But can we use nanotechnology to more precisely deliver agrochemicals, whether it's macro, micronutrients, pesticides? We're working in the space of trying to increase the general abiotic and biotic stress resilience of crops. And when you talk about abiotic and biotic stress resilience, you're talking about climate resilience. We've got some projects where we're looking at promoting either photosynthesis or nitrogen fixation separately or together. In terms of viral infections of plants, we're looking at RNA interference. There are strategies for nano-enabled nutrient fortification. So there's a ton of space in this field where really good science is needed, and that science can come up with really impactful solutions. So the research program at the Ag Experiment Station is kind of laid out here. As mentioned in the introduction, my PhD was actually in environmental toxicology. So when I started hearing about nanomaterials in agriculture, 2005, 2006, I kind of viewed this as an issue of toxicology, that nanomaterials could be kind of the next emerging contaminants, the next PCBs or DDT. So I initially set up a research program looking at the implications of these, and being at an agricultural experiment station, all of the questions were really focused on the potential negative impacts of engineered nanomaterials on agriculture. And got some funding, had some postdocs working in this space. But what we noticed over time, and we published our first paper in 2009, I should say, but what we noticed over time was that there were certain instances where not only were engineered nanomaterials not toxic to plants, but there were instances, exposure scenarios, where there was actually significant benefit that that could be conveyed. So we published, and I'll show you our first paper here in a second, but we published our first paper in 2016 in this space and kind of set up a program looking at the questions that I just kind of went through. So this area of research really just took off, and both in our own institution, but in the field of science in general. For us, USDA has funded much of this work, but I'm actually involved in two centers that are funding this. And this Center for Sustainable Nanotechnology, that's actually why I'm at the American Chemical Society meeting. We're having our final meeting for this center. This center is a $43 million 13-year center, and it's ending this summer. So one of the things that I've pointed out here, and one of the slides I'll show this, is the difference between a nanomaterial having a negative impact on a plant and having a positive impact is very narrow, and much of it is mediated through reactive oxygen species homeostasis. So we're actually taking advantage of some of that right now with some projects. But as I mentioned, this was our first work in this field. We actually began working on soil-borne fungal diseases. These are particularly difficult diseases because the pathogen's in the soil, and it's very difficult to remove that pathogen from the soil. At least in the United States, decades ago, what we used to do is we'd lay down high-density polyethylene and sterilize the soil. Thankfully, we don't do that anymore. But soil-borne fungal diseases, in the U.S. alone, we're losing $200 million worth of productivity. I tend to think about it in terms of lost food, and that's in spite of the fact that we're spending $600 million on fungicides for control. So how we got into this is we know that many of these micronutrients are really important parts of plant secondary metabolism and defense. But it's actually really difficult to get these micronutrients to where they're needed. If you have a soil-borne pathogen, you need these micronutrients at higher concentrations in the plant roots. But when you add these micronutrients by a foliar application, at least in conventional form, very, very little of it makes it to the root system. And when you add them to the soil, obviously they become bound. So we just asked a very simple question: well, what if we started adding these micronutrients in nanoscale form? We know nanomaterials are more active. So that's how we got into this. And we do like to point out that although we are working in disease systems, we're actually not targeting the pathogen directly. What we're doing is we're targeting the inherent ability of the plant to defend itself by stimulating its immune system. So our first paper here was in 2016. We published in NES Nano. It was a combination of greenhouse and field trials with two crops. A lot of agriculture in Connecticut and New England and the United States is focused on specialty crops, such as eggplant and tomato. So we started there. We also started there because we had two good disease systems. So I'll go through some of this data fairly quickly. But what we decided was we looked at 3 different micronutrients, and copper ended up being our best performer here. But we did a foliar application to seedlings, which is how this happens in agriculture in Connecticut. Plants are started in a greenhouse and then transplanted to the field. So we did a foliar application of about 100 milligrams per liter, 100 ppm. It sounds like a lot, but actually we're only adding 1 to 2 milliliters. So the milligrams of each is very low. But we looked at both the conventional forms, the bulk and the salt, but obviously the nanoscale form. And the short of the long here is that this nanoscale copper, which is shown here in green, and the nanoscale forms in the middle bar in the green, we've got increased yield, so increased fruit per plant. We get significantly decreased disease, and we get significantly increased copper in the root system. So we kind of got everything that we wanted. And because this was a field study, we were actually able to do some calculations. We spent about $44 on commercial nanoscale copper oxide. And in so doing, got a $10,000 return in economic yield. But as I tend to prefer to think about increased food production. So I mentioned the Center for Sustainable Nanotechnology, the meeting I'm at now. This is an NSF chemistry center. So we're really focused not so much on increasing yield and agricultural output, but in understanding how we could tune the chemistry of our material to see what was happening at, say, a leaf biointerface. Now, obviously, that has downstream effects, which is why the work's so impactful. But in this case, in this study, this was one we published in 2018. we took our commercial copper oxide and we compared it to a material we made where we changed the morphology and we changed the counter ion. And when you do that, you get significantly different chemical and physical behavior. But what you also see is significant differences in your impact on the plant. So again, this is a foliar application. This is watermelon with fusarium. So this is our measure of disease over here. So this is our disease control with a high rating. And what you see in the yellow bars is our commercial copper oxide. So you got to get up to a couple of hundred parts per million to start seeing the yellow bars decline in terms of disease or increase in terms of yield or biomass. But what you see, the dark green bars are our custom nanosheets, where at a 10-fold lower concentration we're getting that maximum impact. So again, as a chemistry center, being able to tune your materials like this ends up being really significant. As I alluded to, we tend to think about a lot of what we're doing now in terms of stress, whether it's biotic stress or abiotic stress. This is a paper we published in Nature Nanotechnology at the end of 2020, where we really kind of nailed down that mechanism. We started working with soybean and soybean sudden death. We used a whole range of different nanomaterials, including some that we were making in our center. So soybean sudden death, this was just a greenhouse study, but we saw 60 to 70% reductions in biomass and just about every other parameter we could measure. But the application of nanoscale copper was able to reverse this damage significantly. And how we were able to nail down our mechanism was this is really our first effort where we started doing some significant gene profiling. So we're fully adding copper, but we're seeing a change in the gene expression of all of these defense and health related genes in the roots of these plants, which is translating to all of the benefits in the presence of this disease. So why we ended up getting this in Nature was actually the magnitude of the plant response was a direct function of the different types of copper, the different properties. And we were able to actually describe all that computationally because the center that we're in has computational chemists. So another material we're working a lot with is sulfur. Here again, we're looking at the chemistry of the material. Can we add different coatings to alter the impact? You always have to have your conventional controls because you should not be using engineered nanomaterials unless you absolutely have to, unless there's a benefit there. And a lot of times there will be. So we did 3 separate studies, as you can see here, on nanoscale sulfur. The 2 greenhouse studies were actually significant because the postdoc who was working on this, who's now a staff scientist with me, she did time-dependent measurements of just about every parameter that you could think of, including time-dependent gene expression and metabolomics. So we're really able to nail down the mechanism. But here you see kind of the broad impacts, this disease. So this is tomato and fusarium, where you're losing 85% of your biomass, but we're able to alleviate much of that. And again, here's the field study part of that. We're looking at spending about $33 per acre on nanoscale sulfur. And we're taking our yield in a healthy system from about 5,000 kilograms per acre to nearly 12,000. So that is a significant increase in food production, and also having a doubling of yield in the disease system. So some of the other materials we're working with in our center is silica, specifically mesoporous silica with different coatings. One of the coatings we're looking a lot at is chitosan. With the silica work, we're also doing some seed treatments because the material is very amenable to that. But this is the kind of data that you see, you get because of the nanoscale form, you're getting increased concentrations of the nutrient in the plant, and that's giving you all sorts of downstream benefits. And the extent of those benefits is directly related to the chemistry of the materials. So this was another exciting one because in the field study, again, about $19 per acre, and we're producing 20,000 pounds of additional food. And because this is the chemistry center, this is a paper we published in 2019, you can really see how we're focusing. So this is the mesoporous silica with the coating, and this is the uncoated form. And this is kilograms of food produced, kilograms of watermelon. So you're seeing just by tuning that chemistry, you can really maximize your impact. So one of the things that we wanted to find out, this is a paper we published in 2021, was how important is the dissolution of this material. So we're putting the nanomaterials on the leaf surface or in the seeds, but a nanoparticle of silica is basically useless to a plant. I mean, the plant needs silicic acid and usable form. So that dissolution is really important. So a graduate student in our center made 5 different types of silica nanoparticles that were completely identical in every feature, except that they dissolved at different rates. And what we found in this study was that it was the fastest dissolving silica that had the most impact, which is really interesting because if you just add silicic acid, which doesn't have to dissolve at all, you get no impact. So clearly the nanoscale form is really important here. You have to start at nanoscale form, and in this case, the fastest dissolving particle had the impact. But again, here's our field study. This is again with watermelon and fusarium. You know, in the field, we're getting an 80% increase in yield compared to untreated plants. So this is a paper one of the PhD students put together in 2023. Where we really wanted to kind of lay out a tutorial on how you would approach designing a nanoparticle for sustainable agriculture. And we did this because we started seeing a lot of papers in the literature where materials that were being used and there wasn't a significant understanding of the material itself or the impact it was having. So we kind of put this together that would walk you, if you were new to this field, this would walk you through how you would, you know, pick the properties of the material that you would want to try and tune, and you know what questions were we really trying to answer? What was your route of treatment? What was the treatment type that you were trying to address? And then these are the list of questions that you should really be thinking about if you wanted to really design a material and have an impact in agriculture. So this I think was an interesting paper. So this is kind of highlighting nanopesticides specifically. This is a paper that a group of scientists at the Environmental Protection Agency put together, and they asked me to get involved. So we're actually doing an update on this now because this was published in 2022. But they looked at the literature on nanopesticides, and I know I'm running short on time, so I'll pick up the pace here a little bit. But basically what this literature review found at the time was that when you took a pesticide and you added nanotechnology somehow to that formulation, in general, you got an increased efficacy of about 30%, including nearly 19% in field studies. And importantly, that greater efficacy also came with greater precision. We're seeing significantly reduced damage to non-target species. So we're doing a follow-up also looking at nanofertilizers, and to be honest, the data on fertilizers looks even better. So some of the other areas that we're working in phosphorus delivery. A lot of our work on phosphorus delivery addresses the central problem with phosphorus, which is that farmers put phosphorus in the field and much of it becomes unavailable immediately in soil, or some fraction of it washes off. So our strategy here is let's take all of the phosphorus that plants need and let's put them in biopolymers. We're using a whole range of different biopolymers, but these are sustainable materials. We're actually pulling a lot of them from agricultural waste, and it's very tunable. So you can control the rate at which phosphorus is being released. This was from our first study where we showed about a 90% reduction in leaching or runoff of phosphorus from the soil. So it's giving you much greater control. So we have several USDA grants looking at that with different collaborators. We're also working a lot on nitrogen fixation. Specifically, we're looking at biological nitrogen fixation. So this nitrogen is interesting because 78% of the atmosphere is nitrogen gas, but it's completely unavailable to most species of life. But there are a limited number of microorganisms that are able to make that conversion of dinitrogen gas to usable nitrogen. And that happens in a symbiotic association with legumes. So we're really looking to take advantage of that process and try to enhance it. And a lot of this work's actually focusing on nanoscale molybdenum, which is another micronutrient, but it's critical to several of the enzymes involved in the nitrogen fixation process. And much of the work we're doing here is actually focused on the fact that this system, biological nitrogen fixation, is actually very sensitive to environmental stress, sensitive to climate change. So so we're looking at different ways to try and kind of make this system more robust. So we've got a big grant right now that's under review at USDA to look at this. We're also looking at enhancing photosynthesis. If you can enhance carbon fixation, you're making more food directly. So a lot of this work is also focused on nanoscale micronutrients that are specific to the electron capture or the electron transport system, or to the conversion of carbon dioxide directly to sugar. And again, this is a couple of grants being funded by USDA. And the interesting thing we're seeing here is that the carbon and nitrogen cycles are actually very tightly linked. So if you increase nitrogen fixation, carbon fixation increases, just as a function of trying to maintain that balance for species that do both. I mentioned this work on ROS homeostasis. So this again is really looking at in climate resilience in general. So we know that reactive oxygen species at too high concentrations are obviously very damaging, but at lower concentrations are very important signals. And when we look at this, we're approaching this two ways. We kind of laid this out in the Nature Food article that you see here. So you can use nanoscale micronutrients to help a plant respond to a stress, to help it alleviate the ROS either directly by quenching or indirectly by increasing these oxidative stress pathways. But the really interesting work is where we're looking at this preventative side of the cycle. So that's over here. So what we have found, and this is data with rice and rice blast, that you can do a seed treatment or a foliar treatment of rice with nanoscale iron, for example, and that stimulates all of these defense pathways, and then that rice can be exposed to a stress later. So you're basically priming these seeds or seedlings for future hazard. And so this is the unstressed rice. And then these are the different stress treatments. So this is the level of disease from rice blast. So just about any way you do this seed priming, leaf priming of the seedling, you're able to significantly increase the resilience of these plants to future stresses, which is a very significant response. And we haven't published this part yet, but we actually are collecting the seeds from these treated plants, and then this stress memory seems to exist where the F1 generation actually still seems to have significant ability to respond. So I'll just finish up with a couple of things. So in the United States, all of the nanotechnology research funded by the government comes through the National Nanotechnology Initiative, and the National Academies actually is required to do every 4 years to do a review of that program, and that review happened last summer. And one of the things that they do in that review is they try to identify future areas where nanotechnology is going to have a significant impact on both science and society. And this past summer, June of 2024, they identified nanotechnology in agriculture as one of those significant areas for future impact. So I was invited to come down and speak on some of the work that we're doing. So just to kind of finish this up, there's obviously significant interest in this field. Before COVID, we were actually talking to a number of companies, but there was a lot of interest, but no real next steps. But after COVID, this kind of changed. So, we have active funding with at least 3 or 4 major agrochemical companies. We've actually formed our own company called Convergent Bioscience. We're trying to get some funding for that. So I think what's happened here is that the science has matured. I mean, this is the number of publications in this space. But what's also happened is I think many people are now recognizing that, that light at the end of the tunnel, that's a train and it's coming right at us. That's a metaphor we use in the United States. I mean, this inability to feed the people on the planet it exists, and it's getting worse because of a changing climate. So we really need to transform how we're approaching this issue of agricultural production. So hopefully I've convinced you that nanotechnology has significant impact here. Our approach is to start at the start. In other words, understand your mechanisms of action, and that's how you're actually going to have the greatest impact. So I presented a lot of work in a short period of time. We have a large number of collaborators, including a number of collaborators in India. I've been to India 3 times in the last 3 years, and including as recently as October of last year. So I think there's plenty of impactful science here that we need to get done so that we can start to address this problem. So I think I went a little bit long, so I'm going to stop sharing and see if there are any questions.
Thank you, sir, for such a wonderful talk. Sir, the Professor Shahi has joined, or we proceed to the next speaker?
I do not see my picture there, but can you hear me?
Yes.
Yes, sir, we can hear you.
Yes.
Okay, but somehow the video is not working. Thank you very much, Jason. It's nice to see you after a long time.
Sir, give me just
Yes,
2
how are you?
minutes so that I can
Very
introduce
well, very
you.
well. Say again, what you said?
No, sir, please continue, sir.
I'm
please.
so sorry, sir, I'm just interrupting. Can you just give me 2 minutes so that I can introduce you to our audience?
Okay.
Okay. So we have an honor to welcome our distinguished scientist, an accomplished academician, and a pioneer in the field of nanobiotechnology, Professor Shivendra Shahi. Currently a faculty member at St. Joseph University, USA, has made remarkable contribution to the field of biological fabrication of nanomaterials and their potential application. His research in nanotoxicology, particularly in evaluating the environmental impact of titanium, gold, and silver nanomaterials, has been widely recognized. With over 155 publications, 8,623 citations, and an impressive H index of 50, his work has had a profound impact on the global scientific community. His accolades includes the Outstanding Researcher Award, the Career Professional Award, and the editorial position in esteemed journals such as the Journal of Plant Physiology and Biochemistry and the International Journal of Environmental Science and Technology. Furthermore, his contribution extend to patented innovation in the eco-friendly synthesis of gold nanoparticles. Today, we look forward to hearing Professor Sahi gaining insights into his groundbreaking research and exploring the exciting frontiers of nanotechnology and its applications. His presence here is truly an inspiration for all of us, and I am sure we will benefit immensely from his expertise and wisdom. With that, let us all extend our warmest welcome to Professor Shivendra Sahi.
Thank you very much.
Thank you, sir.
So, okay, so Durgesh, we have the list in your pamphlet, that those are correct list, right?
Yes,
The next
yes.
one is Dr. Renato Grill, right?
Yes, sir. So, sir, first I think we have to, sir, as being a session chair, first of all, we have to conclude the lecture of Professor Jason. And after
Okay.
that, if any student have any question, they can ask, they can discuss with the Professor Jason to resolve their queries. Then we will go to the next speaker.
Okay.
So, sir, please, we need your remarks and for Professor Jason talk, and then we will discuss the question answer of the students, if any.
Sure.
Now, sir, your session over to you for your remarks.
Okay. Great job, Dr. Jason White. It's basically you covered the whole area of food nanotechnology, that, it is a growing area, and, lots being done, and especially in your lab and in your collaborators' lab, lot being done and being published. The one question I had is that, has it been taken by the farmers? That some of the technology you are talking about, has it been given to the farmers to try in their field? Hello, Jason, can you hear me? Okay.
Professor Jason.
Apparently he's gone. Okay. Okay. So it seems that, he's not available. So if there is any question, I guess you send me and I will send it to Jason and, try to get the answer for that.
Okay.
So maybe Durgesh, you can try that, or you can send me and I can, I know Jason personally, so try to get those answers.
Okay. Sure.
Yeah.
Sure.
Sir, no problem. The students can write the query in the chat box also. I think then Professor Jason can comment on that.
Now Jason is available. Now Jason is now available.
Oh, hi, Jason. Sorry about that.
That's okay. My cell phone rang. I had to take an issue at the office. So sorry about that. I just had to step away.
Yeah. So my question to you was that you have done excellent in the area of food nanotechnology, and of course your collaborators are doing well. You have published a lot. Some of the techniques that you discuss about that, increasing the food quality and quantity, that has it been sent to the farmers to try in their field, or we are still in the process of developing it?
I'd say we're in that transition phase right now.
Okay.
I think in a lot of countries, this technology is much closer to being in the field, or is in the field. Certainly in China, I think is one of the places, and Renato, I think, is going to be talking about some of the work that's happening in Brazil. But in the U.S., there's definitely interest. All of the large agrochemical companies are now, pursuing research in this area, either themselves or working with, groups such as ours. So I think we're just about there. There are some commercially available products in the United States for farmers to use. I have some doubts about some of those products because I think some of them were produced without an understanding of what was actually going into the formulation. But I think that's happening right now. I mean, we talk to growers all the time throughout New England, and there's tremendous interest from them in this.
Sure. Any other question to Dr. White? Okay. So, Durgesh, should we move on to the next speaker?
Yeah, I think we should. Thank you so much once again. Thank you so much, Jason, for giving us your valuable time for our conference. Your lecture is truly inspirational for those researchers who are working in the nanotechnology. I'm also working in the nanotechnology. So I'm very happy to see your all presentation. I'm following you from last 12-15 years and I have also published several papers with you under your guidance.
Yes.
So thank you once again for your time and your actually we have some 5 to 6 presentations after all 3 talks, in which students I've gone through their work. Students have worked on the the question Professor Sai has asked about the field experiment. What are the impact of nanomaterial on the field level? So some students will definitely they will present what are the impact of nanomaterials on different different crop varieties. So I think they will also give some insight and some fruitful results, so that we can also move forward in the future how we should proceed through the nanotechnology, through the nanomaterial, through the nanofertilizer. Thank you once again, and I hope you will give your more time for this entire session. Thank you so much. Now,
yeah, I'll stay. I can stay on for about an hour, and then I'll have to hop off.
Okay. Thank you so much. Thank you so much.
Okay, Jason. Take care. Thank you.
Thank you.
Okay, the next one is Dr. Renato Grillo.
Yes.
And he comes from Brazil, Sao Paulo State University in Brazil. And I don't have the topic, so I cannot talk about the topic. So is he still? Oh, here. Hi, Renato. How are you?
Hello, Professor Sai.
Hi.
How are you doing?
Very well, very well.
Thank you.
Sir, just give me the brief introduction of Dr. Grillo.
Yes.
We are delighted to host a distinguished scientist, a pioneer in environmental nanotechnology, and a dedicated academician, Dr. Renato Grillo. Dr. Grillo is an associate professor of biochemistry at Sao Paulo State University, Brazil, with an outstanding academic and research background. He earned his PhD and MS degree from the University of Campinas, followed by postdoctoral research at the International Iberian Nanotechnology Laboratory, Portugal, and the Federal University of ABC, Brazil. His extensive expertise lies in developing nanostructured materials for controlled pesticide release, studying nanomaterial interaction with natural colloids and conducted exposure and risk assessment. With over 114 publications and an impressive 10,960 citations, Dr. Grillo has made significant contribution to advancing sustainable nanotechnology application in agriculture and environmental sciences. He is also an inventor and co-author of patents, book chapters, and scientific reviews, playing a key role in bridging research and real-world applications. His reputation as a scientific reviewer for high-impact international and national journal further highlights his influence in shaping the field of environmental nanotechnology. Dr. Grillo's work continues to drive innovation in safer and more sustainable agricultural practices, ensuring environmental protection and food security. We are eager to learn from his expertise, insights, and groundbreaking research. With great enthusiasm, let us all extend our warm welcome to Dr. Renato Grillo. Thank you.
Thank you very much, Dr. Sharma. Very good evening, very good morning for everyone. Thank you for this opportunity. I'm very happy to be here. Actually, it's always a challenge to talk about nanoagrochemical after Professor Jason White. Thank you very much for the very nice presentation. And so I'd like to thank you for this opportunity again, especially grateful to Professor Tripathi for the kind invitation and all the organizing committee. And my name is Renato Grillo, and today I'm gonna talk about nano-enabled materials for the control release of agrochemicals. This presentation will cover the global agriculture context, type of nano-agrochemicals, some examples of organic nanoagrochemicals, inorganic nanoagrochemicals, hybrid nanoagrochemicals that we have been working here in Brazil, some conclusions and acknowledgements. So the global population is growing and is expected to reach around 9 billion by 2050. Additionally, estimated suggest that the food production will need to increase by 70 to 100% to sustain this growth. However, the climate change adds significant pressure of that, with extremely heat in some countries, such as India, wildfires in the US, and floods in South Brazil, for example. These events threaten the agricultural sector and food security. So in addition, as you know, the use of conventional agrochemical is widespread, but their efficiency is quite low. For example, up to 60% of applied nitrogen is lost to the volatilization and leaching. Micronutrients can become invaluable in soil, and only 0.1 to 10% of agrochemicals reach their final purpose due to soil sorption or physicochemical instability. In this way, nano-enabled materials have emerged to enhance the quality and efficacy of agricultural practices, offering potential solutions for increasing crop production, reducing resources used, and minimizing environmental impacts. Application in nanoagriculture include the development of nanosensors, soil conditional materials, seed coating, nanoformulations, nanofertilizers, and nanopesticides, for example. So in this presentation, I will focus on some example of nanopesticides and nanofertilizers developed by our group here at Unesp, São Paulo State University in Brazil. So I divide this presentation into 3 steps. I will show a couple of examples of organic nanoagrochemicals, such as lipid and polymeric agrochemicals, inorganic nanoagrochemicals, such as metal and metal oxide nanoparticles, and hybrid materials which compound organic and inorganic compounds, such as a mix of polymeric and the metallic nanoparticles. So in particular, organic nanoparticles can encapsulate active ingredients such as pesticides, herbicides, and fertilizers within their core, allowing for counter-release as shown in the figure on the right. And you can see here that the conventional active ingredients is released quickly, whereas the encapsulated one has a slow release profile. So this behavior can promote several advantages, such as protection from photolysis, increased pest control efficiency, protection from biodegradation, reduced deletion and volatilization, and of course, control release of pesticides. So in this study, we developed chitosan tripolyphosphate nanospheres to encapsulate the no-selective herbicides, paraquat, using the ionic jellification method. Paraquat, as you know, has high water solubility, but the encapsulation efficiency reaches 62% in our system. Chitosan nanospheres had average size of 208 nanometers, and the nanoformulation showed a counter-release profile in relation to the no encapsulation system, as you can see here, and enhanced the herbicide activity was observed in plants. In another study, we used the potential mechanism of action. We evaluated actually the potential mechanism of action of this nanoformulation using in vitro and simulation tools, and it was observed that the distinct electroactivity behavior of the nanoherbicide changed the interactions between the ferrodoxin and ADP reductases. So this is a catalytic domain is involved in electron transfer process of photosynthesis in plants. Another examples involves the development of lignin nanoparticles for the counter-release of plant hormones. Auxin was used as the active ingredients model in this study, and the lignin was sourced for cellulose paper factories here in Brazil. So the lignin nanospheres had average size of 120 nanometers and demonstrated a good colloidal stability. And the counter-release profile of nanoformulation is lower compared to the non-encapsulated activity ingredients. And interesting, exposure of lettuce seeds to lignin nanoparticle resulted in large plants with increased chlorophyll content after the germination. Also, lignin nanoparticle exhibited antifungal properties with improved germination profile when this nanoformulation was used for nanopriming of seed, for example. So here we present some results on lignin nanoparticles for the control release of herbicides, atrazine. Atrazine was encapsulated in the system with an encapsulation efficiency of approximately 92%. The nanoparticle exhibited good colloidal stability with a diameter of 200 nanometers, and the formulation demonstrated enhanced herbicidal activity in a quite chemocraft compared to the conventional formulation as well. So the most interesting aspect of this nanoformulations is that we simulated all the chemicals in separated boxes using molecular dynamics simulation, as you can see here. Over the time, we observed the same spherical morphology as in the scan and electron microscopy characterization. It's a very interesting because simulation can aid in the production of safe by design nano-enabled agrochemicals, semi-chemicals, and enhancing the development of smart or intelligent nanostructures. And now I will present one example of metal agrochemical. In this study, we developed a magnetic nanoherbicides to encapsulate usnic acid, a liquid metabolite with herbicidal activity. And different magnetic nanoparticles were produced here with and without oleic acid capping. This formulation exhibits good colloidal stability as well, particularly for the oleic acid coated this kind of nanoparticles. So the size distribution of this nanoparticle was 15 nanometers, very small particles. Additionally, this magnetic nanoherbicide was tested against weeds, and it was observed that both free usonic acid and usonic acid loaded magnetic nanoparticle exhibited herbicidal activity, but nanoparticles shows more effective than free usonic acid in inhibition of the photosystem 2 of the plant. So to finish, I will show you two examples of hybrid nanoagrochemicals that we have been producing here in Brazil. More recently, we have been developing supraparticles for agricultural applications. Supraparticles are self-assembly structure in a unique system, and then we can form it from multiple nanoparticles without the use of polymeric surface, as you can see here. For example, you can produce different kinds of supraparticles. For example, this one is a zinc oxide supraparticle. The system has potential to combine different nano-enabled materials into a single system and promote a slow release of activity of different active ingredients. In this graph, you can see the in vitro control release of zinc from the supraparticles and the conventional nanoparticles in water. And it is observed that the supraparticle have a lower release rate than the zinc nanoparticles, the dissolution profile of the ion. This approach is particularly interesting because it allows for the modulation of nutrients release and soil by adjusting the properties of this kind of materials. In addition, supraparticles can reduce nutrient leaching in the soil. We have exciting exciting finds about this, and enhance chlorophyll content in mice as demonstrated in our study. In this example, I'd like to show another type of hybrid formulations that we have been developing here in our lab. These are biodegradable polymers hydrogel spheres. It's not a nanomaterials, but we can encapsulate different kinds of nanoparticles, organisms in the systems, provide protection for molecules and organisms against abiotic and the abiotic stress in the field. We can produce spheres with different kinds of materials, such as xylem, bentonite, cellulose, lignin, alginate, and so on. Here we have bacteria in nanoparticle encapsulated in polymeric hydrogel. I think that I don't have time to present these finds now, but I hope to have the opportunity to discuss them in another time. In general, these spheres protected the bacteria for environmental degradation and enable the control release of nutrients from the nanoparticles. It's a very interesting system. So, as you can see, some of the results presented here are interesting. However, nanoagrochemical face significant barriers to overcome in the coming years, such as it's interesting to talk about scale-up formulations to deliver at field scale, construction of new nano-enabled agriculture materials, more precise. Would the nano be the best choice, or maybe a supraparticle or something like that? It's important to talk about regulation and safety concerns and consumer perception as well. Sorry.
Thank you, sir.
In conclusion, no, I have it. A couple of
Okay. I'm so sorry, sir. I'm so sorry.
Okay, in conclusion, nanotechnology has the potential to dramatically improve the agriculture and help feed the world. However, its application must be safe and sustainable, of course. This is part of our facility at UNESCO. We have a range of analytical instruments, for example, DLS, NTA, AFM, scanning electron microscopy, and so on. Additionally, we have available experimental station located 5 kilometers from our department. And of course, we are open for the collaboration. Here, I'd like to invite you to submit a paper to the special issue on plant nanobiology that we are organizing. The submission deadline is here. And so I would like to thank you, all the institutions and colleagues who participated in some of our studies. Also extend my gratitude to the funding agents such as FAPESP, CNPq, and CAPI, especially FAPESP. And of course, a special thanks for our students who are always dedicating the lab. So Ilha Solteira is located in the northwest region of São Paulo state in Brazil, while it does not have ocean beaches, but you can see both several stunning rivers, beaches surrounded by beautiful natural landscapes and birds, as you can see here. So thank you for the opportunity. Thank you for your attention. This is my email, and thank you.
Thank you, sir.
Thank you so much, Dr. Grillo. Excellent presentation. Excellent, using nano for the organic as well as inorganic content to, of course, to look the effect of that on plant growth and development. One of the question I had is, did you guys see that, what would be, let's say that, if you're trying that in the field, and what would be the long-term impact of these nanoparticles in the field? I mean, to the same generation as well as the crop being produced in the future.
Okay, Professor Sahi, thank you for your question. Wow, very good. I have difficulty to respond to your question because there are a lot of parameters that involve the ecotoxicology or something about nanoagrochemical. So I think that the first step to answer your question, I need to think about the biodegradable. I think to produce biodegradable nanocarriers using, for example, lignin nanoparticles or some cellulose or something like that. And also, I think that it's important to use different kinds of agrochemicals. For example, natural agrochemicals like essential oil or something like that. And also, it's improve, I think, this field to enhance the studies about the mechanism of action to properly full response your question. It's important to understand the size, to understand the coating of the nanoagrochemicals, to understand the interaction. Of course, we need to change the standardization, the methodology to evaluate, to study nanoagrochemicals in different kinds of methodology. I think that's okay. I don't know if I answer your question.
Well, that we need to think about one thing, that that whatever composition we are using, that should be biodegradable. Somehow it is going to be degrading, so there may not be any long-term effect on the crop, right? Okay, great. Any other question, anybody?
Yeah, yes, Professor Sai, I would like to just discuss with Professor Renato. Very good presentation, and so much of novel addition in nanotechnology. I don't have any question, but I just want to discuss two things. One is about the shelf life of the nanomaterials which you have prepared, and we are suggesting to apply in the agriculture field or in the lab. So what about the shelf life? The first thing. So second is when we encapsulate, because I have seen several of your papers, and we also you are my very strong collaborator. We have done it. Means published several papers. We have recently started in my lab about the how we encapsulate nitric oxide and indole acetic acid other than any phytohormones. So what is the future if we encapsulate these means gasotransmitter as well as some phytohormones? So what is the future of these kind of nanoparticles? How we can because initially we have just started in my lab. So what is the future of these kind of nanoparticles we are encapsulating, and what the nanoparticles which you have prepared and applied, what is about the shelf life?
Okay. Professor Tekpachi, thank you so much. I'm very happy to be here. It's a pleasure to see you again.
Thank you.
Of course, we have a lot of collaboration and works that we have done that I would like to work with you and your group. So about the self life. Okay, we have some structure that the self life is like months, for example, 2 months and 3 months. But we have other nanoparticles, especially lignin nanoparticles, for example, that the shelf life is more than 1 year in water, in dispersion. It's a very interesting. Of course, we can dry this formulation, but sometimes we have problem to disperse these nanoformulations because the physicochemical stability changes. So it's need to be careful to do that. But yes, we have a good shelf life of these nanopesticides that we have been produced here. Also, about the encapsulation, I don't have experience in oxygen nitric or something like that. I think that Professor Halley has a long experience about that. Especially we encapsulated here organic pesticides or essential oil, organic molecules. Also inorganic species like zinc or iron oxide and so on. To encapsulate this kind of to encapsulate this kind of compounds, you need to have very good design nanoformulations. For example, you can encapsulate the hydrophobic system in nanocapsules. It's okay. You also can encapsulate it metal nanoparticles or iron or something like that in nanospheres. For example, you don't have the oil core, so you need to think about the applications before they constructed the nanocarriers. About the future, actually, we have some experiments here in soil column, and especially nanoparticles in soil. We have great lixiviation as well. So if we can construct the supraparticles, it's like different kinds of nanoparticle in a big one. I think that you can release the active ingredients, you can release the nanoparticle without the lixiviation process, for example. So I think that you need to, there is no the special point, but you can change different formulations to the final applications in soil. Maybe you can nano to spray in leaves. Maybe you can construct a small nanoparticle to track in the plants and so on. So thank you very much, Professor Tripat, for your question.
Thank you so much, Renato. Thank you so much for coming and joining this session. I'm very thankful to you. Now, I would request you to please be in this session because some students will also present some field work and some 4 to 5 presentations. So thank you so much. Now over to Dr. Sarma, please.
Thank you, sir.
Thank you, Renato.
I would now welcome our next speaker for the session, Dr. Modolo, a renowned scientist in plant biochemistry and molecular biology, whose work has significantly contributed to the field. Dr. Modolo earned her MSc and PhD from State University of Campinas, Brazil, and pursued postdoctoral research at a Nobel Research Institute, USA. She further expanded her expertise as a visiting professor at the University of North Texas. Currently, she is an associate professor at the Federal University of Minas Gerais, UFMG, Brazil, focusing on plant secondary metabolism, nitrogen nutrition, and stress responses. With over 110 publications, 23 book chapters, 4 scientific books, and multiple patents, Dr. Modolo is a leader in research and innovation. She serves as associate editor of the Journal of Advanced Research and editorial board member of Plant Nanobiology, actively shaping the scientific discourse. Beyond academia, she is the founder of G Bioplan, and co-founder of RedNew, driving research on plant biochemistry and urease inhibitors. Her collaboration spans Denmark, Egypt, and the USA, India, Italy, and she is an active member of leading scientific societies. Dr. Modolo's dedication continues to inspire the future of plant science and sustainable agriculture. We are delighted to have you for this session, ma'am.
Thank you very much. Can you all hear me?
Yes, ma'am, you are audible.
Yes, Professor Majuro, yes.
Oh, that's good because my computer, the mic is not working. I needed to set my cell phone and the video comes from my computer. The voice comes from my cell phone. I'm glad you can all hear me. I'm going to share my
PPT.
My screen.
Yeah, we can see your presentation.
Okay.
Perfect.
Okay. First of all, I want to thank Professor Tripathi for this invitation. And just to put in context that I am an infant here because I'm just starting to work with nano-based products. But I want to share with you what we have done so far in the scope of agriculture and how can we combine what we have done with nanoparticles, for example, in order to get encapsulated and better results in the field. So then, therefore, I'm going to just focus on the climate change as well, because this is something that we need to keep in mind. With the increase of a world population, the demand for food is going to increase considerably. But we also need to face the fact that we impose pressures in the ecosystem. And as a result, this will considerably trigger and accelerate the process of climate changes. And of course, the plants will suffer from that because the productivity will generally go down. When we think about the Malthusian theory, in which it is stated that if we keep producing food with the current technology and we don't invest in new technologies, we are going to reach the shortage in a few years. And as a result, we are going to reach food security. And we need to make plans to overcome this situation. And one of the main goals of my research group is basically to develop of new sustainable and environmentally friendly technologies to make plants to benefit from what they can get from the soil system and improve the fertilizer efficiency and also the crop yield and in turn the food quality as well. When we think about nanotechnology, I like to highlight about nanoparticles in specific because they pose as a very good systems to improve the ability of delivering interesting compounds, nutrients, and so on. And this is particularly important in the agriculture because we can use nanoparticles to promote the plant growth in a whole, which it is one of the goals of my talk because I work with biostimulants and also to improve the efficiency of fertilizers. Among the methods that are currently developed for the synthesis of nanoparticles, we can see on the screen that one very interesting one is the one based on biological synthesis, in which you can use, for example, microorganisms, plants, and any other biological material to get nanoparticles and in a manner to combine with even metal ions or organic compounds, which it is the case of my group that we are interested in. And we have several good results here regarding the effect of nanoparticles, and I am just highlighting some here on the screen, that it is basic of nanoparticles related to important nutrients or also related to carbon nanoparticles, in which we can find out they will boost the micronutrients uptake by plants, boost the root elongation, seedling growth and germination, root growth and plant development as a whole, and so on. So the aim of my research group is basically to develop urease inhibitors and biostimulants and use nanotechnology to further enhance their effectiveness in promoting plant growth and productivity. And to talk about urease inhibitors, I need to mention about urea. Urea is the most worldwide used nitrogen fertilizer because it bears several advantages, like the high content of nitrogen, the low price per production related to nitrogen atoms, and it is easily to manage on soil. And but the problem is when we just apply a urea on the soil, the extracellular urease present there basically will hydrolyze this molecule, releasing ammonia and CO2. And because ammonia is a gas, it's gonna go to the atmosphere and incur in lots of nitrogen losses. But if this ammonia is basically percolated in the soil and it's going to go very near to the rhizosphere, it's going to be transformed into ammonium due to the presence of water in the soil solution and become the cation that it is readily absorbed by plant roots. And here is a panel just to show you how this negatively impact the agriculture if the activity of urease present in the soil is really, really high. The idea of using an urease inhibitor that will temporarily inhibit the urease present in the soil is just to make sure that we are going to just slow down the production of ammonia gas and in turn prevent the volatilization, because this will indirectly be responsible for the environmental pollution as well. And if this ammonia is nearby the rhizosphere, by the action of water, it's going to become this ammonium ion, and then it will be readily absorbed by plants. If it is oxidized by nitrifying microorganisms, it's going to be converted in nitrate, and nitrate can be also uptaken by root systems, but can also be leached from the soil if it is not properly managed. And also it can suffer the activity of the nitrifying microorganisms and incur in the production of very toxic gases that will directly or indirectly contribute for the formation of greenhouse gases. For this reason, we focus on the inhibition of urease as a manner of coping with the nitrogen losses and also the environmental negatively impact. We have several approaches to do that, and we focus basically on the urease inhibitors. On the other hand, when we think about biostimulants, biostimulant is any substance or a microorganism or part of it that when you apply to plants, you can enhance the nutrition efficiency, abiotic stress tolerance, and or crop quality traits, because you are going to boost the natural metabolism by plant in order to make them more feasible and eligible to cope with diverse situations. Therefore, fertilizers and pesticides do not fall in this category. Some examples of biostimulants available in the market include humic and fulvic acids, some protein hydrolysates, seaweed extracts, and botanical extracts as well, chitosan, inorganic compounds, and some beneficial bacteria and fungi. When we think about the urease inhibitors, what we do basically in our team is not new. There is a commercial urease inhibitor, it's called NBPT, which bears this chemical structure, which mimics the structure of urea, the substrate of urease. And therefore, this substance works as a competitive inhibitor. The problem with NBPT is it works very good in temperate soils, but in tropical soils like those in Brazil, the stability is very poor, and as well as the shelf life. Some examples of urease inhibitors include silver nanoparticles, quinones, coumarin derivatives, and thioureas substituted ones, and several phosphoramidates, all found in vitro inhibition. But when we move to the soil system, we notice that the variety of compounds that are eligible to inhibit urease, it goes down. And when we go to the plant soil system, this is still narrowed because we sometimes cannot validate what it is found in the lab or even in the soil. Then there is a challenge to slow down urease activity. Our group basically focus on the nature as inspirational source of compounds that are eligible to inhibit urease. And our primary focus is on modifying natural products in order to get better urease inhibitors that will effectively decrease the nitrogen loss to the atmosphere and also make more urea available for the plant to be uptaken in the form of ammonium, but just when the hydrolysis is done in the rhizosphere. Okay, so now I'm going to show you some examples of good lead compounds that we got so far. And that we want to do this to encapsulate with nanoparticles in order to make more readily for the plant. Thinking about phenolic aldehydes, we noticed that when we tested this urease on soil, we found out that, for example, vanillin and seringaldehyde are two examples of phenolic aldehydes widely distributed in plants, but they are not very good urease inhibitors. But when we did some molecular modification in the core of this phenolic aldehyde, we got two derivatives that were very efficient and were able to inhibit the enzyme by 60%. Protocatechuic acid is another example of a phenolic aldehyde that per se is able to inhibit the urease activity by 50%. But when we did extra changes in the core of this structure, we were able to get more efficient compounds, derivatives that were able to basically inhibit up to almost 100%. And when we checked this in the millet soil system, we found out that plants that were treated with urea in the presence of one of these compounds basically were able to grow faster and accumulate much more biomass. And the relative growth rate was really increased by this particular derivative when used at 0.5% weight in relation to urea. Another example is another type of phenolic compounds that we checked, and we found out they were very good urease inhibitors, except for the resorcinol that was a poor inhibitor. And they were as effective as the NBBT. But for our good surprise, when we checked these compounds for the phytotoxicity by using the lattice system of seed germination, we notice, I'm sorry the screen is not very good here, but believe me, the root system here, when treated with hydroquinone at 100 micromolar, it is bigger when compared with the control, which shows us that besides the ability to inhibit urease, this compound is also interesting for boosting the root growth, acting therefore as a potential biostimulant as well. And we notice for a methylated compound also improved the root system growth at a concentration that was half the one of the unmethylated compound. And when we check it, for example, for one other class of organic compounds that we did a modification in some amino acids, we used basically the amino acid as a core for the chemical modification. We noticed that two of the compounds that we got, they were very effective in inhibiting urease in soil, as effective as the NBBT. And when we checked this in the soil millet system, we were very surprised in a good way. I mean, we were expecting that, but when we confirmed that the amount of nitrogen in plants in the shoot portion of the plant considerably increased, and phosphorus as well, and potassium, and magnesium, and sulfur, and iron. And then we were pretty excited because besides the NPK increment, because they are demanding by plants in higher amount. And we noticed that, for example, the iron content was also increased, suggesting that if we use this compound in a formulation containing urea, we can basically improve the urea efficiency. And just for the sake of numbers, because numbers are more striking, we noticed that the nitrogen content in the shoot was basically 24% higher when compared with plants that were solely treated with urea. Phosphorus increased by 66%, sulfur 34%, and magnesium 41%. The root biomass of those plants also increased by the presence of these two compounds that we called root buster because of that. And they were very effective in boosting the root growth and also biomass accumulation, while NBPT failed to do that. And it is noteworthy pointing that NBPT was not effective in improving the uptake and the transport of those nutrients to the shoot. So then, by investigating a potential phytotoxicity of these compounds, we noticed that among 5 of eligible and very good compounds that effectively inhibit urease, we noticed that 3 of them, highlighted here as RB3, 4, and 5, were able to improve the root length by 100% on average. And while 2 of them were not so effective when compared to these 3. And these results are quite striking because when you compare the treated seedling, you see that the root is way bigger than that from the control plants. But notably, when we further evaluated the RB1 and 2, we noticed they were not able to improve the root growth, but they were able to improve the formation of lateral root, which it is very interesting because when you think about the root architecture, both the main root and the lateral roots are very important for the uptake of water and soil nutrients, which tells us that, for example, we can basically combine these 2 types of compounds, derivatives, and get a formulation that would boost the plant root growth at the most. We show here this panel just to demonstrate that the shoot part of the seedlings were not negatively affected by these compounds. And the yellowish color here in the plate is just a result of the color of this compound, which it is produced in a single reaction, okay, and then in high purity. So, to summarize, basically what we have noticed is the design of a new urease inhibitors and biostimulants will basically potentially improve the nutrient uptake and transport. And we hope it will also improve the assimilation because this is something that we are pursuing right now through experiments that are ongoing And as a result, we believe that some crop traits like the sugar content, the fruit color, and nutrition will also improve, because the presence of these compounds in a formulation will boost the plant metabolism and therefore the yield. And we have some evidence that I just didn't bring here because we are just treating the metadata that we got it about the influence of these compounds on the soil microbiota. But some evidence we have that they don't disturb it and can improve the performance of some rhizobia. And also, we believe that in this sense, because the root system is increased in size and in architecture, we believe that the plants will be able to cope against some abiotic stresses such as water deficit, and because it will improve the water efficiency. All of these could not be possible by a very good team of students that currently are just hold their PhD. I want to notice Ms. Andrea Silva, Dr. Oliveira, Dr. Duhaniki, Dr. Soares, Dr. Horta, Dr. Chaves, Dr. Avila for their efforts to gather these results. And I also want to thank my colleagues in this big project, because we are part of a National Institute of Science and Technology in urease inhibitors of agricultural and medicinal interest, in which I coordinate the agricultural part and Dr. Di Fatima coordinates the medicinal part. Dr. Goulart has a very important role in this team because she is responsible for the formulations. She has a very good background in pharmacy, and she basically tells us what adjuvant and what kind of solvent we can use in the system to make sure that for the regulatory policies will be met, and then we can further pursue this and eventually one day get a product in the market. And also want to acknowledge Dr. Mahiel, because he is the microbiologist, soil microbiologist in our team, and he basically works with us dealing in this matter of the soil and the relation with the plant. I just want to point out that all of these, basically, these compounds, they are scalable, which it is very good. We are in phase of producing in the order of a kilogram of them. And in order to basically get the best system of nanoparticles to encapsulate it for testing in the field. I just want to express my gratitude to the committee of the 8th AMIFOST and my institution, Federal University of Minas Gerais, the NIST and urease inhibitors, and also the network of biostimulants and fertilizers of improved efficiency, and the financial support from CNPq, FAPEMIG, CAPS, and Embrapa Milho Sorgo. Thank you very much, and sorry if I just extrapolated my time.
Thank you, Dr. Modolo. This is Shivendra Sai here. It was a nice presentation, very well done. And that I don't have a question, but I have a comments to make, and especially for those students, that they are going in this area, in the nanobiotechnology area, and also to the young researcher, that nanotechnology is multi-discipline area. You can be an expert in physiology, plant physiology, biochemistry, molecular biology, but you cannot be expert in all these areas. So you have to essentially develop a contact or working relation with other people, because being in one area you cannot do nano biotechnology. Thank you. Durgesh, any other
Indeed.
question?
Totally agree with you.
Yeah.
Professor Bujia, thank you so much for your outstanding presentation. And I've gone through your entire presentation. You are expert in urea, application of urea in plant biology, and how they are benefited to the plant system, yield production, and the quality and quantity of food production. Recently, I don't know if you have might be heard about the nano urea, which has been launched by one of the very well established company, IFCO, of Indian company, which has launched the nano urea, which is very much popular nowadays. And farmers now they are utilizing the nano urea in the agriculture field, and they are getting so much exciting results about the how nano urea can be utilized instead of the bulk urea. So you can look into in this direction also. In the nano system, we are also working in this area. So overall, your a very nice presentation. And now another concept is nano biostimulant. You are an expert of biostimulants, and I'm connected with a very long duration of time with you are my collaborator now. You are also the part of my plant nanobiology initial board. So I know about your entire work. Nanobiostimulant is another concept people of all over the globe around among the research field. They are also working in this direction, nanobiostimulant. So thank you once again for joining us in this a very good session and giving your valuable time. Now I would like to hand over this session to Dr. Sarma and Dr. Monica Thakur, Professor Monica Thakur, and Professor V.K. Modi sir, Director of Amity Institute of Food Technology and also the chair of this session. And Professor Monica Thakur is the convener of this AMIFOST, very renowned international conference. Every year, it is Amity Institute of Food Technology is organizing every year. So it is now a brand of our Amity University. It's a very prestigious. So now I would like to hand over the session to all the organizing members of this international conference. Thank you so much, Rajita. Thank you.
Thank you, Dr. Durgesh. And especially this session was conducted as per Indian time. It is a little bit late, but anyway, it was so interesting that we all enjoyed, and I'm sure that all participants must have learned the basic thing about this, the concept or the importance of the nanoparticles in the agriculture field. And especially when one of your student, Dr. Durgesh student, is working on this, and now student is from food technology, but he's working on this concept, and I'm seeing the results. It is a really means very, very interesting, and means it may change the concept of agriculture in the future, that especially with the use of fertilizers and all, what everybody is claimed that we use excessive fertilizer just to protect our crop, but nobody is looking after the health of the soil, health of the plant, and then health of the fruit. And ultimately it comes to the human chain system. So that is the most important part. But we are now looking for this, that yes, everything should be healthy, environment as well as the health of the consumer. Ultimately we are the consumer, and parallelly of course the plant, the consumer is the animals also. So overall we have to look into this concept, that when we use the nanoparticle for the agriculture purpose, especially in the form of pesticides, that the safety of this everything that must be taken care. And I'm sure that all the scientists in this area, they are aware about this, and as going through the presentations, yes, I'm very, very, very much impressed that yes, This is the area where it is now, it is coming very fast. This is, I think, going to be just like your genetically modified food. And now the label will come that nanopod crop is grown through nano fertilizers. It is already there, I think, in the commercial use, but it is still not has become so popular. Anyway, I thank Professor Shivendra Shahi, Professor Johnson, Professor Renito, and then Professor Luzia. They have spent their time, and with the help of Dr. Durgesh and Dr. Monica, you are here with us, and I'm very happy that you could spare some time and share your all the scientific knowledge with all our students and participants. And this particular session, which was included in our AMIFOST, that is also a credit to the conference. Yes, a very renowned scientists were there for this particular, and it's a new concept for our conference. Earlier, we were not using this concept. We were using mostly for the post-harvest technology, for the processing or products or whatever it is in the food technology. But this is, of course, for this AMIFOST, we introduced a new concept. So I'm thankful to all the speakers, and so we'll take it further for the next conference also. Thank you. Thank you, Dr. Durgesh.
Thank you so much. Thank you, sir. Thank you so much.
Yes, Dr. Sunayan, please. Dr.
Thank
Monica.
you, sir. Yes, sir. Okay. Ma'am, I'll ask our convener, Dr. Mona Thakur, to please share her thoughts.
Yes, again, good evening, everyone. So once again, thank you for all the distinct speakers of the day, and specifically Professor Shivendra Sahi sir. I think he has taken time from his valuable time from his very busy schedule. I know it's very difficult for him because to join and then to listen to every distinct talk and then express his views and then again chairing the session, very difficult for him from such a busy schedule, but still he's with us and still he's means listening to every talk and then expressing his views. Then special thanks to Professor Jason White, Professor Renato, and Professor Luzia. So we are very fortunate. I think we listened to all the talks very carefully, specifically all the students who have presented their work, presented their abstract for this specific session, that is harnessing nanobiology for agri-food technology. I think they have listened to many of the thoughts, many of the new things they can add. And again, thank you, Dr. Durgesh. So we are very fortunate that this session is being conducted as a part of the AMIFOST. Thank you once again. I think we can start with the oral presentations now.
Yes, ma'am. Yes.
Thank you once again. Thank you, sir.
Thank you. Thank you so much, ma'am. Thank you. Now we should move towards
so I would request, yes, sir. So we'll be starting with the online oral presentation for this session. And I request Ms. Vishakha Sharma and Akriti Srivastav to kindly start presenting their work on the topic impact of salinity stress on wheat production, a growing concern of food security for the growing world population. Vishakha Sharma, Akriti Sharma, Akriti Srivastav, sorry.
Ma'am, am I audible and visible?
Yes, you're audible and visible as well. You can start sharing your slides.
Dr. Sunayan, please also advise student about the time limits, so that they can finish within the time of 5-minute presentation, I hope.
Yes.
Okay.
And 2-minute
Sir,
questions.
yes. Sir, give me 1 minute, please. Yes,
Yes,
I'll announce. So there, all the participants, all the oral presenters. So the time limit is only 5 minutes. You have to bind up your work in 5 minutes, and then it will be followed by 2 to 3 minutes of question answers. So I request you to please share your screen and then start the slideshow mode. Yes, Vishakha, please start.
Durgesh, can I listen to their presentation?
Yes, sir, definitely.
Yes,
Yes,
sir.
Yes, sir.
Yes, sir. It will be our pleasure if you will.
Yes, sir. It's an honor for us. Yes, sir. It's an honor for us, sir.
Sure. Thank you.
Thank you, sir. Thank you so much.
Vishakha, I request you to put your slide on slideshow mode. Yes, you may proceed. Vishaka, please unmute yourself. You are muted.
Vishaka, there are two different buttons giving on this thing, the extreme left hand side. One is for mute, second is for stop video. I think you have muted yourself. Please unmute yourself.
Is it disabled at your end? Okay. I think you need to see your mic.
Visakha, please unshare your PPT. We till then we can invite another student. Please resolve your technical issues. Till then we can invite another student.
So now I can unmute myself.
Yes.
Sharing my mic got mute. Let me share it once again.
No, please share. Yes.
Fine. Am I audible?
Yes, you are audible. Vishaka, you may proceed.
Okay. So, a very good evening, everyone. My name is Vishaka Sharma, and today I'm going to present my MSc dissertation research work. But my abstract that I have submitted for this conference was on different topic. So the topic which I'm going to present right now is about evaluating the efficacy of biochar nanoform on the growth of cadmium stressed wheat. So coming to the background of my research work is cadmium is one of the non-essential and non-biodegradable heavy metal considered as highly toxic environmental pollutant worldwide, causing serious environmental and agriculture problems. Cadmium contamination in agriculture soil negatively affect the crop production by inhibiting the plant growth, reducing the yield and potential accumulation in the food crops. Nanotechnology has shown the promising potential to promote the sustainable agriculture. In this study, I investigate the role of carbon nitride nanobiochar on the growth and development of wheat morphological parameters under cadmium stress. So coming to the methodology, what I have opted for, first is the surface sterilization of the seeds with 80% ethanol. Then I have transferred them in muslin cloth for the further germination. Then after 3 days, I got the sprouted wheat seeds. Then the germinated seeds are then transferred into Petri plates and grown in the Hoagland solution. These Petri plates are then kept in the growth chamber for about 1 week with 65% relative humidity and 25 degree Celsius temperature. Then after 1 week, uniform size seedlings are selected for the treatment. These were my treatments. T1 was control, T2 cadmium under the toxic concentration of 100 micromole. Treatment 3 was carbon nitride nanobiochar. And treatment 4 was the combination of carbon nitride nanobiochar and cadmium. So here figure number 6, it illustrates about the uniform size wheat seedlings. They are transferred into the cups and treated with different treatments. Figure 7 illustrate the morphological impact of the different treatment in the wheat plant. So these were the data analysis techniques or instruments I have used. So first, for the measurement of the root shoot length, I have used the centimeter scale. For the measurement of fresh and dry weight, I have used analytical weighing balance. For the then the plants are kept in the hot oven for around 4 to 5 days for the dry weight measurement. And I have also cut the root sections of the wheat plant to and observe under the microscope. So this was the graphical result I have obtained. So there were various morphological parameters. First parameters is about the root length. So as you can see that when the plants they are treated with cadmium, the root length was reduced by 36%. as compared to control. Whereas carbon nitride nanobiochar, it increased the it increased the shoot length by 31% as compared to control. However, the combination of carbon nitride nanobiochar and cadmium, it also increased the shoot length by 16% as compared to control. Coming to the second parameter, that is the root length, similar results were obtained. That is the toxic concentration of cadmium, it reduced the root length by 99% around, and the carbon nitride nanobiochar, it increased the root length around 51% as compared to control. Whereas the combination treatments, it increased the root length by 24% as compared to control. Shoot fresh weight and root fresh weight, I also obtained the similar results. That is, shoot fresh weight, in the shoot fresh weight, the toxic cadmium concentration, it reduced the shoot fresh weight by 32%, and the carbon nitride nanobiochar, it increased the shoot fresh weight by 64% as compared to control. Whereas in the combination treatment, I get the results that the increase of the shoot fresh weight was increased by 27% as compared to control. Similar result I get with the root fresh weight, as you can see the graph and my table. That the toxic concentration of cadmium decreases the root fresh weight by 24% as compared to control, whereas carbon nitride nanobiochar, it increases the shoot fresh weight, the root fresh weight by
You have 2 minutes
30%.
more, Miss Visakha. 2 minutes more. Please finish
By
your
30%. Yes, sir. By 30% as compared to control, and in the combination, it increases by 25%. Similar result I got for the shoot dry weight and root dry weight. You can see the graphical representation as well as my table. I also did the in vivo visualization of the histochemicals. So figure A, it illustrates about the impact of different treatment on the membrane damage of wheat. And the figure B, it represent the impact, illustrate about the impact of different treatment on the lipid peroxidation of the wheat plants. So, in conclusion, I can say that this study highlights the significant role of carbon nitride nanobiochar in eliminating the harmful effect of heavy metal toxicity that is here in the case of cadmium stress, in the case of cadmium. And this research illustrate how carbon nitride nanobiochar at its beneficial dose, how it eliminate the toxic effect of cadmium on the wheat growth by increasing its morphological attributes. However, to verify the results, the field experiment should also be conducted for the food security and safety for the consumers globally. So these were the references. Thank you. Should I unshare my screen now?
Can we
Hello?
ask question? Can we ask question?
Yes, sir, definitely, sir.
Okay, hi Vishakha, very well done. My question is that, your conclusion is good, but I would like to add in hydroponic situation because you don't know, it has not been tried in soil. Have you tried in soil?
No, sir. My study was on the hydroponic basis only.
Yeah,
That
so
is why in
just put it
conclusion I have added.
Yeah, just put it there. If somebody sees the conclusion that, may not understood that, that it is under hydroponic situation. Okay, because you don't know that how it will affect in case of soil, unless you do some soil study in the greenhouse. Okay.
Sure, sir.
Good job.
Ms. Vishakha, I have also one question. The entire data which you are showing, in hydroponic, yes, sir, is Professor Sai sir is definitely the suggestion you should consider that you have to also means initiate your experiment in the soil condition, as well as you have not. I have gone through your all figures. You have not put in the figures the standard error. And also one of your figure in which you are showing that when you combine your biochar along with the along with the cadmium, the concentration means the increase means the growth is increased 18%, which is not possible because under cadmium stress you are showing 36% reduction, and when you include your nanobiochar, the concentration should be the reduction rate should be decreased, not, but you are showing 18% enhancement. So I to the best of my knowledge, it's not very much possible. So you are suggested to go through your data again, analyze it very carefully. The rest you have done a good job. So thank you so much.
And you show that data. Show that there is a
please
error bar.
show your data.
Show that data.
Yeah, this one.
Yeah, there is there was a error. Oh, error in the table. If you see error, what is that error means?
Yes, sir. But in figures, error is missing, and the shoot the table in which the shoot length data you can see, sir. C is showing 36% reduction in under cadmium stress, and under carbon biochar C is means the data is showing 31% enhancement in the growth shoot length. But when he C combined along with the cadmium, the enhancement is 16%. So I don't think you should, in my view, you should analyze your data. 36%, so maybe reduce at 20% or maybe 12%, 16%, but it's not possible that when you apply your carbon nanobiochar along with the cadmium, and you are going to show that carbon nanobiochar is more capable, and it can nullify the means the toxicity of cadmium. In my view, and to the best of my entire knowledge and experience, I don't think it is possible that any material can nullify more than 100% of the toxicity. So please analyze your data, Ms. Bisakha Sharma. Thank you so much. Now you can unshare your PPT.
So I call upon our next presenter, Shivani Mahara, Sneha Tripathia, Kavita Tiwari, Shobita Methva, Kritika Singha, and Shivesh Sharma. They will be presenting their presentation on biopolymer-based strategy for enhanced survival and controlled release of plant growth promoting bacteria under environmental stresses, a way towards sustainable agriculture.
Good evening. Am I visible?
Yes, your PPT is visible and you are very much audible.
Good evening, all the dignitaries present here. Myself, Shivani Mehra, and the topic for my presentation is polymer-based strategy for...
Shivani Mehra, I would request you to please put your presentation on.
Shivani, please continue. Shivani, please continue.
Yes, sir. The topic of my presentation is biopolymer-based strategy for enhanced survival and controlled release of plant growth promoting bacteria under environmental stress, a way towards sustainable agriculture. as we are facing, as the environment is facing much of the stresses, in which the abiotic stress is the most significant one, like salinity, heavy metal, drought, temperature. There are many conventional approaches through which these stresses are being mitigated, but due to the negative effects like their short shelf life, their frequent application, their rapid dispersion, their environmental sensitive and immediate release, they are reducing the growth, they are leaching out, and moreover the uneven distribution. So there was a need of an hour to move towards the smart delivery approaches, which will enhance the growth, yield, and defense mechanism through the targeted delivery, reduced pollutant toxicity, controlled release, involved soil aggregation, improved shelf life. As encapsulation. Encapsulation is being a very effective formulation that protects the bioactive compounds. That can be any bioactive compound, including PGPR, salicylic acid, nitric oxide, and many nanoparticles. And that will allow the controlled and their sustained release. This is the methodology how it has been prepared. The bioactive compound is being filled inside the syringe, and with the solution with calcium chloride, the dripping ionic gelation method continues and the beads are formed. The layer can be either any polysaccharide, lipid, protein, or it can be any polysaccharide based nanoparticles like chitosan nanoparticle. The choice of carrier depends upon its biodegradability, its biocompatibility, the released carrier residues, its encapsulation and loading efficiencies is a main protocol, their solubility and shelf life, the shape and size of the bead. So why these encapsulation is required? The main reason behind is the controlled release of these bioactive compound from the encapsulated beads, as it can be the targeted delivery at a specific time whenever the release is required to protect the PGPRs from the harmful environment or harmful stresses. The methodology involves the bacterial cell, its centrifugation, then mixing the alginate and starch or chitosan nanoparticle solution in the magnetic stirrer, and the PGPR loaded beads are prepared through the solution. The main role that plays it is calcium chloride solution, which with the bond of which in the contact with alginate produces a gelation method and produces the gel-like beads. The beads are shown like this. These are the results. These are the prepared beads. The beads were measured also. These are the dried beads, and these dried beads were screened for its encapsulation efficiency, its release behavior, biodegradability behavior, and swelling behavior. The encapsulation efficiency.
You have 5 minutes for your presentation and 2 minutes for the discussion. So please compile your presentation within the time frame.
Yes, sir. The encapsulation efficiency, as we can see in the graph, I got the encapsulation efficiency above 90%. These were the beads which were spread out so that I can confirm that my beads were encapsulated within the alginate. These are the pellets which I directly spread plated by serial dilution, and the beads which I soaked in PBS for 1 hour, sonicated it and spread plated it. These were the different efficiency, like encapsulation efficiency, as I discussed, it was above 90%. Swelling efficiency for 216 hours, the swelling efficiency was good, but later on it started degrading. It was in NaCl. The biodegradability, I checked it in the pot with the soil, and the beads being kept in muslin cloth. So I saw that up to 60 days, the beads were almost degraded within the soil. So according to these results, the swelling behavior and the liquid content increased as the starch gelation, chitosan nanoparticles, and alginate concentration did. So 1.5% of starch concentration was taken and 2% of alginate was taken. The percentage varied, as we varied the percentage, the shape of the beads also varied. I went for different types of techniques like X-ray diffraction, FTIR, TGA, scanning electron microscopy, and TEM. These are the references. Thank you.
Is there any questions?
Shivani, I have one question for you. Actually, you have showing the capsulation. So can we fix into the nano form, because these are not in the nano form? The
Yes,
size is very large. We cannot say that the particle which you have prepared and you are going to utilize in the agriculture field for the safer food production, the quality and quantity food production. You would also like to you are also suggesting the nutrient quality of the food. So can we fix these particle into the nano form, means in the nano shape?
Yes, that can be referred as the loading, and then we will be checking for the loading efficiencies. So we will be getting the mixture as in the form of precipitate, but not in the form of beads. In the form of beads, it can be gone into nanofam. But if we want to encapsulate with nanomaterial, so we will be having the loading efficiency, not the encapsulation efficiency.
Okay, so what will be the difference? What do you think? What will be the difference? Means these nanoparticle and nano, when you means prepare these part into the nano form, what will be the difference? What are you expecting?
Sir, the application part would be different. It will be easier if it is made in the form of precipitate, so it will be easy to give it in foliar form. So, but the bead part is important for PGPR, as PGPR are mostly required in the soil. So we want that treatment which will be benefiting it in the soil. So that's why we are making the beads. So it has a control and slow release, and it's being less affected. As if we do the foliar spray of PGPR on the leaves, it is of no use, and it can be directly affected through the external environment. So that's why main target was to cover up the PGPR with a suiting material.
Okay, perfect. Professor Sai sir, would you like to ask anything?
It looks okay. The only concern I have is if you go to one of the slide, you're showing certain days, but you need to, I'm talking about in terms of publication. If you send it, they are going to say that you start with 0.5 or days 5 or something like that. One of the slide over there, that try to do it 0. Okay.
Oh, okay.
Because, yeah. So it will be consistent, because you don't know when you took it, how did it look? Those kind of thing.
Yes, sir. From 0 interval itself.
Yeah. You had several of those. You start with certain number, but have a control as a 0 as well. Okay.
Okay, sir. Thank you, sir.
Shivani, I also have one query. So
Yes, ma'am.
question, but one query that I think, have you done the comparison between the precipitate and the alginate, like the foliar application and the soil application? So is there any comparison done, or?
Ma'am, my junior is working on this precipitate form itself. So yet we are working with the foliar thing, as we need any solvent also to dissolve these precipitates for spraying it in.
Okay.
But it's tough to encapsulate this PGPR in the precipitate form.
Okay.
So, ma'am, but other chemicals like nitric oxide, we can salicylic acid or IEA, these kind of phytohormones or signaling molecules, we can do it in the form of precipitate form.
Okay. Thank you.
Okay, thank you. Thank you so much.
Okay, so we move towards our next presenter, Shobita Matthew, Shivani Mehra, Kavita Tripathi, Sneha Tripathi, on nano encapsulation of indole 3-acetic acid, a sustainable innovation for targeted delivery in agriculture to enhance food security, plant health, and stress resilience.
Hello.
Yes.
I'm audible?
Yes, Shobita, you are very much audible.
Okay. Thank you. Good evening. I'm Shobita Matthew, a PhD scholar from the Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad. So I'm pleased to deliver my presentation on the topic nano encapsulation of IAA loaded phytosan nanoparticles, a sustainable innovation for targeted a sustainable innovation for targeted delivery in agriculture to enhance plant health, stress resilience, and food security. So, giving an introduction to the topic, plants as sessile organisms are affected by various environmental stressors such as light intensity, drought, temperature extremes, herbicide toxicity, various oxidative stresses, nutrient imbalances, heavy metal pollution, etc. All these affects the morphophysiological and biochemical and molecular processes in plants and finally leading to yield loss. The data here shown highlights a critical situation with various abiotic stresses causing reduction in crop yield, making them the primary constraint on crop productivity. So, according to United Nations Organization, global population is projected to reach 9.8 billion in 2050. This is leading to an increased demand for food. To meet food security, we need to adopt strategies that enhance agricultural productivity. So, in this light, my study is significant as it focuses on the sustainable crop improvement and stress mitigation with the application of nano-enabled bioactive ingredients. Why nano-encapsulation? So, traditional formulation, which is used as agrochemical formulations, used are affected with various drawbacks such as low efficiency, low solubility, low stability, low utilization rate, non-target effects, low bioavailability, high chemical load on environment, and burst release. And also, if we directly apply on soil or in the agricultural system, it releases rapidly and leach out. It causes soil toxicity also. While in the case of nanocarrier formulations have improved efficiency, increased solubility, low toxicity, high stability, high utilization rate, high bioavailability, and low chemical load on environment, and they act as smart delivery systems which can be used for controlled release of the active ingredient. So this slide shows the mechanism of controlled release. In conventional systems show a rapid release, and nanocarrier systems have the controlled release. And there are various mechanisms by which this controlled release acts. Various mechanisms such as photodegradation, hydrolytic degradation, pH stimulated degradation, and enzymatic degradation. So here is an outline of my work on IAA nano-encapsulation. IAA is a phytohormone which has proven its efficiencies in crop improvement and plant growth promotion. So firstly, chitosan nanoparticles are dissolved in acetic acid solution, and this suspension of chitosan TPP nanoparticles loaded with IAA obtained by inotropic gelation method. It is centrifuged and the chitosan nanoparticles loaded with IAA is recovered. After that, the recovered nanoparticles are lyophilized for storing, and we have calculated encapsulation efficiency and loading efficiency using these following equations. So, encapsulation efficiency refers to the percentage of an active agent that is successfully encapsulated within a carrier material related to the initial amount of the active agent used in the encapsulation process, whereas loading efficiency represents the ratio of encapsulated compound to the total weight of the carrier system, indicating how much of the carrier is occupied by the active ingredient. As you can see in the graph, chitosan nanoparticles loaded with 8 milligrams of IAA exhibited higher encapsulation efficiency and loading efficiency, making them optimal choice for further analysis and investigation. After that, I have done characterization of the synthesized nanoparticles. I have done particle size analysis, swelling study, FTIR, TGA, X-ray diffraction, and TEM. So the particle size distribution was, the data was obtained using particle size analysis. The chitosan nanoparticle loaded with IAA, their size ranged from 12.71 nanometer to 832 nanometer, and the swelling study showed that the encapsulated carrier system has a high swelling efficiency, which shows that chitosan is a good biocompatible, and it makes the IAA encapsulated within the system more bioavailable.
Ms. Shobita, please follow the limit of the timing. You have 2 minutes more to finish your presentation.
Okay, sir.
Okay.
Then the 10 studies showed that the nanoparticle size ranged below 20 nanometer. Release studies showed that cytosine nanoparticle loaded with IA can act as a controlled release carrier system. And FTIR and X-ray diffraction data showed that the nanoparticle IA encapsulated within the carrier system has high conjugation within the cytosine TPP, and the presence of IA specific peaks, hydrogen bonding interactions, and all these FTIR analysis data showed that IA is encapsulated within the nanoparticles without disrupting their core structure. So, in conclusion, these findings suggest that IEA-loaded cytosine nanoparticles have significant potential for stress mitigation in plants. So, the future studies will focus on further exploring their effect in various adverse conditions in crop plants. Thank you.
Any questions from the speakers and from the participants?
Ms. Sobita, I have one question for you about your presentation. You have presented excellent data about how you have encapsulated indole acetic acid. So application part, I think, is still missing. You have not applied these encapsulated nanomaterial into the plant system.
Sir, I'm working on that, sir. This was the first objective, so I have completed that.
Okay, so
what
you are expecting, if you are, because we all know that indole acetic acid is itself is very effective against the any abiotic or biotic stress if we are utilizing into the means as a bulk material, right? So why you think that it is important to encapsulate it? What kind of impacts you are expecting if you will utilize encapsulated indole acetic acid in place of
actually, yes, sir. Yes, sir. You are right, sir. IA is very efficient phytohormone, but it has some drawbacks such as it is very toxic to plants if applied at once at high concentration, and also it is light sensitive and it is heat sensitive. So it has these drawbacks, so that we have to apply it frequently. So it's a major drawback. So by encapsulating, we can avoid frequent application and encapsulated higher amount of IA. Plants can grow by taking up smaller amount that is released upon demand kind of mechanism, and there is no need to change. If we are since I am working on hydroponics, I have faced that the change this encapsulated system. If I am applying this encapsulated IA, I don't have to change the whole solution every day. But in the case of direct IA application, I have to change the solution every day since it is degrading so fast. And also it is protected from photosensitivity. And the plant will also get the combined effect of cytosine and IA, since cytosine is also acting as a biostimulant itself.
Okay, actually, Sobita, the first thing is that there is no need to change every day. If you are going to utilize indole acetic acid in hydroponics, it's not necessary to change every day the concentration of the solution of indole acetic acid, right? It is you can change into after 272 hours. The last one question is that we all know that plant has its own mechanism to control the production of any material, including the phytohormones. Yeah, they have a control mechanism. Because you know that endogenous biosynthesis is a process through which plant always utilize the auxin, and they utilize this phytohormone for their growth and development, right? So the toxicity in the natural condition, toxicity is not we cannot say that toxicity, because of toxicity maybe plant feel indole acetic toxicity, right? Plant has its own mechanism to control. Yes, we are agree. Then if we are utilizing the nanomaterial into the encapsulated form, it may be beneficial. But the most important is in front of all of us, that what is the shelf life of your nanomaterials. If you are going to say that somebody, means farmer can apply this encapsulated nanomaterial, indole acid cap nanomaterial, then what is the shelf life? So you should also work in this direction to increase the shelf life, as well as you should also first apply these encapsulated nanomaterials and bulk material, and you should see what is the overall impact. You should compare the nano encapsulated indole acid nanomaterials and the bulk material. Thank you so much, and
Yes,
best
sir, sure.
from my side.
Thank you so much, sir.
Thank you.
Thank you. I call our next presenter, Akriti Srivastav, and she would be presenting her PPT on zinc nanoparticles, modulate arsenic absorption, and ROS scavenging, which are key components of arsenic and NaCl mediated cross tolerance process in rice.
Miss Akriti, please share your PPT. Please unmute yourself first.
Hello?
Presentation.
Yes, Akriti, you are audible.
Ma'am, you can see my presentation now?
Yeah, we can see the presentation. Please start.
Okay. Okay. Good evening, everyone. Myself, Akriti
Akriti, 1 second, Akriti, please stick to the time. You only have the 5 minutes, okay? Please stick to the time.
Yeah, ma'am. Yeah, ma'am. Okay, okay. So, my topic for today's presentation is zinc nanoparticles play an important role in arsenic and NaCl mediated cross tolerance mechanism in rice through modulation of arsenic uptake and ROS scavenging. Before going into the topic, first I would like to discuss about what are the problems that India is facing worldwide. India and the worldwide is facing in agriculture, and what are the solution to these problems. So, talking about soil and water contaminations in India, there is various abiotic and biotic stresses which affect the soil and as well as crop in India adversely. So, about the abiotic stresses, arsenic and salinity is one of the huge problems that worldwide is facing. Arsenic is highly toxic metalloid adversely affecting the environment, human health, as well as crop productivity. Arsenic concentration in the soil in India range between 3.34 to 105 mg per kg soil, indicating the damaging effect on health of human and as well as plant. Hence, it need a control that arsenic toxicity by developing reliable and cost-effective methods. Talking about the salinity of soil, it is also one of the major problems that agriculture is facing worldwide, and high salinity level causes adverse effect on soil properties and production of the crops. So here are the global distribution of salt affecting land area and also distribution of soil affecting land in India. So impact impact of salinity on soil, it impact morphologically, physiologically, and molecular also. It reduce the yield and productivity of the crop. It also reduce water uptake, so and also damage the plant cell under salinity stress. That's why the plant cannot grow properly, and it damages the whole plant growth. And also the import and impact of arsenic from soil to rice grain. It also arsenic affect the rice grain and damage the rice grain of the crop. So to overcome this problem, the stresses, there are advanced technology, that is nanotechnology, by which we can improve the crop production and meet the growing global demands for food, feed, and fuel while practicing sustainable agriculture. In controlling condition, nanotechnology as an essential material generally benefit for plant yield, even at the high concentration, but at a certain concentration, at a high dose, it also possesses toxicity. So among all these essential metals, zinc oxide nanoparticles, a micronutrient, has reported as one of the most beneficial nanoparticles for the plant life. So further, the role of zinc oxide nanoparticle on regulating various aspects of plant metals, what are their aspects, we'll discuss further in my treatment. So these are my treatments I have done while doing my experiment. I have taken control arsenic, salinity, zinc oxide nanoparticle, arsenic plus NaCl as a cross tolerance, and arsenic, NaCl, and zinc oxide nanoparticle, in which we can clearly see that the arsenic growth is lesser in all because it's a toxic, but NaCl growth is higher than arsenic. But when we see in arsenic and NaCl in a combination form, the arsenic salinity mitigate the effect of arsenic. The study assess the synergetic effect of salinity and arsenic on physiological and morphological parameters, indicating the potential metabolic pathway involve cross tolerance of arsenic with salinity. So by giving the morphological analysis, we can clearly see that the fresh weight of rice seedling under the treatments, the zinc oxide nanoparticles give the maximum growth, but when we keep the arsenic and NaCl together, the growth has been increased as compared to arsenic and NaCl. Same as with oxidative stress marker, we have taken the oxidative stress marker as a superoxide radicals, in which we can see that arsenic level is high because of the accumulation of ROS in this. Same with the hydrogen peroxide in rice seedlings, the arsenic level is higher as compared to different treatments. MDA, milanaldehydes. And now we can see the root section of rice seedling in the membrane damage, in which we can clearly see that arsenic has much accumulation of oxygen in the cell sections. And this is the data of the lipid peroxidation. The same arsenic has the maximum damage in the root section. We have nitroblue tetrazonium result of root tips. This is the root tips, H2O2 hydrogen peroxide. We have DAB, and this is the data of the antioxidant enzymes that plant involving various antioxidant enzyme to detoxify ROS, including superoxide radical ascorbate peroxidase. Here's the amount of catalase, and this is GST activities. Here's the glutathione reductase content and its gene expressions. Here we can see the positive result of this. And here showing the proline metabolism in rice under the arsenic and NaCl stress condition exposed to zinc oxide nanoparticles, as proline is the multifunctional amino acid that is accumulated in high concentration in plant under various stress condition, where we can clearly see that the proline content in arsenic is higher as compared to other treatments. And when we taken the arsenic and NaCl together, we can see that the concentration of this arsenic and NaCl as compared to arsenic and NaCl is much lower.
So you have 2 minutes more to finish your presentation.
Okay, sir. So after the hydroponic, we have seen the positive result of arsenic and NaCl and its cross tolerance. We have moved forward to the field experiment also, and we have done the pot experiment after the hydroponic experiment, and we have grown different treatment, different replicate of rice seedlings, and we have seen up to the 240 days of treatment, and we have concluded with the same result as we have seen in the hydroponic condition, that when we applied arsenic individuals as arsenic and NaCl, and we applied arsenic and NaCl together, we see the positive result as you can show in the data. I have in the field experiment, I have taken 2 cultivars of rice seedlings, rice seeds. One is P44 and one is PB150P. The results so same as the hydroponic one. And here also you can see that the zinc oxide nanoparticle rice, after the harvesting, we have taken the rice panicle, in which we have seen that zinc oxide nanoparticle has the maximum rice grain as compared to the arsenic or NaCl or arsenic and NaCl together. And when we have taken arsenic, NaCl plus zinc oxide nanoparticle, we have seen the positive result. Same with the second cultivars. And also we have calculated the grain height and its filled or unfilled, and we have seen the same result as we have seen in the hydroponic. And the size of the grain are significantly different under the exposure of salinity and arsenic. We observe that the flowering and the fruiting is delayed under arsenic and salinity condition, but it has seen the positive result when we have applied arsenic and NaCl together. So we can conclude that the application of nanoparticle improved the size of the grain, weight of the grain, and also significant the manage of the flowering and under the exposure of salinity and arsenic stress. So we have seen the positive result, including hydroponic as well as in the field experiment also, and we are further going with this results. We have. So I conclude I conclude with this result that it indicates the positive effect of zinc oxide nanoparticle on the arsenic and salinity state toxicity in rice plant. And also we can see that when we applied arsenic and NaCl together as a cross tolerance, we can see the positive result when we see individuals. So the antagonistically influence of arsenic and NaCl on absorption of each other of the rice crop has been confirmed. Result confirmed that the zinc oxide nanoparticle confirmed cross tolerance against arsenic and NaCl toxicity seedling and a grain in rice by enhancing the activity of antioxidant enzymes and reduce the amount of oxidative stress marker. So its finding, my finding of this result has an agronomically advantage of maintaining the crop productivity in arsenic and salinity in soil globally as well as in India also. Thank you.
Any questions from the speakers and from the participants?
Yes, sir. Please, sir. Yes, sir.
Yeah, I have a question.
Yes, sir.
This is your PhD project, right?
हाँ जी sir. Yes, sir.
So did you try or do you plan to attempt looking the nutrient concentration in
yes, sir.
in seeds, right?
Yes, sir. Yes, sir.
Yeah, include that one because that will make a nice story. Okay.
I'm going with that only. So I'm doing my work is going with the nutritional quality and analysis of the grains, rice grain.
Okay. Very good. Very good.
Yes.
Actually, Professor Sahi sir is suggesting you to analyze the nutrients, means micro and macronutrient concentration, while you are utilizing the arsenic and NaCl, and when you combine both the treatments, right? So you have to think. I hope you have already given the samples to analysis.
Yes, sir.
I have 3 questions, one by one. You have to answer. First, why you have taken 2 rice cultivars? This is the first question. Second one is, what is the role of antioxidant metabolism you have analyzed? What is the role? And how you will explain when you have combined the arsenic and NaCl toxicity, then what is the role of the antioxidant metabolism? And the third one is, what is the basis of the dose selection of arsenic and NaCl. Have you analyzed it? Have you screened it out?
Yes, sir. Yes,
Yeah,
sir.
so please give your answer one by one. I have asked 3 questions, so you have to
Sir.
be, yeah.
Thank you, sir. Your first question answer is ki, why I have taken 2 cultivars as a rice crop. So there is various quality and quantity of the rice crop different, and I want to see that if different crops had different functions, different growth activities, or it has same. So I want to just take the different, because rice has different varieties.
Have you analyzed in the hydroponic condition the similar as you have means
No, I have not done in hydroponic, because I have to just analyze in hydroponic that what are the result while doing the treatments. Ki it get positive result or negative result in one cultivars the rice have. After that, I have taken in a field the grain. I have to see the grain in the field, but in a hydroponic I can't see the grain, nutrition quality and quantity. That's why in hydroponic I had just seen the result of the
come to the
shoot.
next question, please.
Yes, sir. The next question is arsenic and NaCl together as an antioxidant enzyme?
Yes. How you explain what is the role of antioxidant in the plant system?
I didn't get the question.
Why you have analyzed?
So antioxidant enzyme has analyzed for the involvement of ROS in the plant, the role of ROS.
Come to this the third questions. I think you should rework and means you have to read some papers to take the idea why one should analyze the antioxidant when they are providing any stress. Why the need of antioxidant metabolism, analysis of antioxidant metabolism, and what is the role in ROS scavenging? You should be very means well acquainted with this knowledge, right? The third one, what is the basis of the arsenic and NaCl dose selection?
Sir, we have taken NaCl as a 150 millimolar and arsenic as a 10 micromolar concentration.
On what basis?
Sir, we have we have read from the previous paper, that what they have done already, what the toxic level they have seen, and what are the beneficial concentration already mentioned in the paper. We have taken the difference from that.
Your lab in your lab, it has not been established before. You are totally based on the previous literature, or you have follow your
Protocol. I have taken protocol from my guide. So I have taken the protocol from that only, the previous seniors only. So I have done.
You should answer like this now. Ki your lab is already have screened out.
My lab. We have previous reference that I have not done the screening, but from the previous my lab colleagues only I have taken the previous references.
What about why you have analyzed the proline metabolism? The last question.
So to see the concentration. So to see the concentration of stresses in the plant, the stress condition. It's amino acid.
Thank you. Sir. Any other, if anybody wants to ask the question?
I think, sir, we can start with the next presentation.
Thank you, sir.
I call upon our next presenter, Garima Balian, Akhilesh Kumar Pandey, and they would be presenting on hydrogen sulfide and titanium dioxide nanoparticles work together as to relieve tomato seedlings stressed from salt.
No, I think she has not joined, so I think move towards the next presentation.
Okay, sir. So we move towards our last presentation for this session, Pratyusha Dhal, and she would be presenting on the topic impact of copper and iron nanoparticles on photosynthetic pigments and stomatal frequency of rice plant.
Good evening, everyone. Ma'am, is my PPT visible?
Yes, Pratyusha, you may go ahead and you're audible as well.
Good evening, all the honorable guests over here. I'm Pratyusha Dhal and I'm currently pursuing my MSc Agronomy from Amity Institute of Organic Agriculture. My study is on impact of copper and iron nanoparticle on photosynthesis and stomatal frequency in rice plant. As we know, with the growing global demand, we need to boost the crop productivity, especially for staple crop like
Tusha,
rice.
you have only 5 minutes, so you please follow the limit of the means presentation. Please finish your
Sure, sir.
presentation in 5 minutes.
Definitely, sir. So nanotechnology presents a shift and it gives a boost by manipulating the materials at nanoscale and by potentially revolutionizing the plant nutrition, stress management, and overall crop yield. Among various nanoparticles for my study, I have taken copper and iron nanoparticle due to their essential role in the metabolic processes. Going ahead with the materials and methods. First, I have taken the rice seeds and soaked it and sterilized it and soaked it for 24 hours. And after that, I have kept it in muslin cloth for 2-3 days, allowing it to germinate. After its germination, I transferred it to Petri plates and kept it in the growth chamber. And after its sprouting, I prepared the Hoagland solution. And for going ahead with the treatment, I first prepare the stock solution. And for the stock solution, my treatments are 4 treatments. First one is iron nanoparticle at 25 micromole and iron nanoparticle at 500 micromole, copper oxide nanoparticle at 15 micromole, and copper nanoparticle at 100 micromole. From that, I have made the working solution. And as you can see in the slide, this is the day 1 where the rice seedlings are transferred in cups after segregating based on their size, and we have transferred it in. I have transferred in cups in different treatments. After day 3, we need to change the solution. And this is the day 12 where the rice seedlings are ready to do all the analysis and data analysis for further experiment. As you can see in the result, this is the morphological impact of different treatments on roots of rice plant. As you can see here, at the low concentration, there is an enhancement in root length, there is increase in shoot length, and the root hair is also improved. Whereas in the higher concentration of nanoparticle, the root and shoot length is decreased and the root hair development is not that well. This is the data of the morphological impact. And in this graph, this is the graphical representation of the root length and shoot length. And in this also you can see the root and shoot length shows a positive impact at lower concentration, whereas in the higher concentration, there is a decrease in the root and shoot length. This is the graphical representation of the shoot weight and root weight. Here also we have seen a positive impact in the lower doses of the nanoparticle, whereas in the higher doses of nanoparticle, there is a decline in the root weight and shoot weight. This is the dry shoot weight and dry root weight result. Here also the results are the same, from which we can conclude them in the biomass levels have increased in the low doses of nanoparticle, whereas in the high doses of nanoparticle, it has decreased. I have done the histochemical staining also, where we have of root sections, where we can see there is an increase accumulation of stains in the higher doses, where you can we can conclude there is a lipid peroxidation and skip staining and the membrane damage at the higher concentration of the Evans blue stain. Whereas the root sections are as comparatively better in the low doses of the nanoparticle. In this, I have taken the root tips, and here you can see for we have taken 4 stains, Evans blue. First one is Evans blue, then skip staining, then DAB, and then the last one is NBT stain. Here you can see in the Evans blue.
Piyusha, you have 2 minutes more. Please finish your presentation.
Yes, sir.
Please conclude,
In the higher doses, we can see the accumulation of membrane damage, where the stain has accumulated more in the higher concentration, whereas in both the iron and copper nanoparticle. In this, I have represented the impact of different treatment on stomatal frequency, where I have counted the stomatal count under microscope, and this is the data which I have taken of the stomatal frequency, where you can see in the iron at 25 micromole and copper nanoparticle at 15 micromole have a positive impact. The stomatal frequency is more in that. Whereas in the higher concentration, the stomatal frequency is less. And I believe that maybe this is a planned defense mechanism due to which the plant the absorption level have decreased due to that. Then we have done the extraction and analysis of photosynthesis pigment. First, we have taken 20 milligram freshly weighed leaves, and then we crushed it in 80% acetone. Then the pigments were filtered and centrifuged, and after that, the absorbance was read at 3 wavelengths using spectrophotometer. This is the absorbance at different wavelengths in rice.
Piyusha, please conclude your presentation.
Yes, sir. And this is the result of the determination of total chlorophyll A, B, and total carotenoids, which also shows the positive impact in low concentration and less impact in the higher concentration. So the result of this study demonstrate that at lower concentration of iron and copper nanoparticle, there is improved growth of rice seedlings, and at lower concentration, the photosynthesis pigments like total chlorophyll carotenoids were also enhanced, and the plant biomass and stomatal frequency has also increased. Whereas in the higher concentration, the plants have induced to oxidative stress, leading to a decline in the growth metrics, and it has also lowered the chlorophyll concentration and reduction in stomatal frequency. However, we need to do further studies, because my study is based on the hydroponic experiment, and we need to apply it in the field condition to see whether the farmers will really be able to apply this in the field condition. And we also need to have the consideration whether we don't use so much of heavy metals as overutilization of metal containing manures, fertilizers, pesticides leads to contamination of soil. Thank you very much. And lastly, I would like to say that my study is still going on and I am yet to do the molecular level analysis and study for my experiment. Thank you so much.
Any questions? Any questions from the speakers, participants?
Yeah, I have one question, only one. Pratyusha, you are showing the concentration of stomata, right? You are showing when you are utilizing the beneficial doses of the nanomaterials, the concentration of the stomata increase. They are increased, right?
Yes,
but
sir.
when you are providing the toxic doses of the stomata, they are reducing. What is the strategy of plants? Why they are reducing the stomata under the stress condition, and they are increasing the stomatas in the beneficial condition? And what is the impact of these means ki what is the impact of these increasing and decreasing stomata on the food quality and nutritional quality? How you will connect it, food and nutritional quality of the seeds or the particular plants which you have?
Sir, I believe this is a plant defense mechanism. The plant has to limit the nanoparticle uptake. Plant has limited the absorption, prevented the excessive use of water. That's why the stomatal count is less, to prevent excessive water loss and to reduce the absorption of nanoparticle. And as you said, that how I can relate it to the food, to the other levels. So for that, I would like to answer that I also mentioned previously that we need to do the experiment in field levels, and if it also shows the positive result, then the farmers can apply it in the field level, and it will also boost the productivity. And as we for rice, we can all, as it is a staple food, it will be a great way to increase the productivity.
Thank you, Pratyusha. Thank you.
Any other question?
Yes. Pratyusha, is there any limitation with your work while doing the work with these objectives? Do you find any limitation?
Ma'am, the limitation is that I'm doing it in hydroponics, so there it is in a protected environment, and the temperature and the humidity is also decided as it is in the growth chamber. So if as long as we won't be able to do it in the field, we won't know the proper result.
And whatever experiment we are utilizing the nanomaterial, and the main motive behind is that we are looking towards to feed the world growing population. So we have to also focus on the nutritional quality, nutrient availability, carbohydrate, protein, fat, these kind of things, and macronutrients, right? How so you have to also keep my suggestion when you finalize your experiment, that how you will correlate like the growth parameters, stomata, and photosynthesis and all at all with the main food production quality as well as the quantity food production. Thank you.
Yes, sir. Thank you, sir.
Any other question?
No, I don't have any question.
Okay,
Ma'am, if you would like to ask anything.
No, sir, nothing. Yes.
Okay.
Okay. So we come to an end of our technical session 3. So I thank all the invited speakers, those who have taken time from their busy schedule and joined us for this session online for their Indian Standard Time. I also extend my warm thanks to Dr. Durgesh Tripathi sir for the pioneer speaker for this and being the chair or co-speaker for this session. And lastly, our very honorable HOI sir, Professor Dr. V.K. Modi, for being the chairperson and joining us online. Thank you, sir. The results for this session will be announced in the validity session tomorrow. The session is also, the validity session has been mentioned with the link online, so all can join. And those who can come physically, they can be there in the validity session. So tomorrow the results will be out. Thank you, and have a good day and a good night.
Thank you so much.
Yeah. Thank you once again. Thank you all.
Thank you so much, ma'am. Thank you so much. Thank you so much, Modi sir, and thank you all the organizing team for giving me opportunity to interact with the scientists and students. Thank you so much. It's always my pleasure. Thank you. Thank you so much, ma'am. Thank you so much, sir.
Thank you.
Thank you, sir.
Last में आ गया मैं.
Yes. So we request the IT team to close the session, please. Close the session for the day.
Yes, sir. Okay, sir. Thank you so much, ma'am. Thank you, sir.
Yes,
Thank you.
everyone. Thank you so much.
Thank you, sir.
Thank you. Thank you.